A short introduction before we start. Welcome everybody. We're going to do this discussion. Julio from Oviedo, Jose also from Oviedo, and we have we should actually wait until exactly.
She gets bad reception where she is. She gets bad reception where she is.
Because I can't see your screen actually. It's because you use this. Sorry for the interruption.
All good? Okay, I'm going to check this with. Okay, great. Hi everybody, my name is Farley Miller, and I'm presenting my thesis work, which is Seaweeds, a natural approach to improve ruminant productivity, quality of animal products, and reduce environmental emissions. To do my research, I went to the Queen's University of Belfast, which is in Northern Ireland, in the United Kingdom. I joined the Institute for Global Food Security, which is run by Professor Sharon Hughes. My supervisor was Dr. Katerina Theodorodou, and I joined the larger PhD project by Kaylee Barnes, which is part of the Sea Solutions group of projects, which is looking at marine solutions to agricultural problems in the United Kingdom. As this is the only talk you're going to see today about cows, I'm going to go into a lot of background. We'll talk about global livestock production, focusing on Northern Ireland, the present and the future. We'll talk about the environmental emissions from those practices. We'll talk about enteric fermentation, which is cow digestion. We'll talk about some options for reducing those emissions, and then we'll get to the beef of the study, which is using seaweeds as a feed supplement to reduce the environmental impacts. Globally, animal products compose about 21% of the calories consumed by humans. As quality of life improves around the world and people have access to healthier and more varied diets, this number is expected to increase in the future. Focusing on Northern Ireland, these are the greenhouse gas emissions from all sectors over the past 30 years. You can see a downward trend across most sectors, except for the green line on the top, which is agriculture. Agriculture is a notoriously difficult sector to decarbonize. The United Kingdom has a plan to reach net zero greenhouse gas emissions by 2050. Right now, Northern Ireland is not on track to meet that goal. These are the projected greenhouse gas emissions in 2050. The majority of which in the middle are from agriculture, and the vast majority of the emissions from agriculture are methane from enteric fermentation, from livestock production. Enteric fermentation in its simplest is cow digestion. As cows consume plant material, it enters the rumen, which is the largest chamber of the four-chambered stomach. The rumen contains a diverse microbiome composed of fungi and archaea and bacteria, and they ferment or digest the cellulose plant material into energy for the host animal, in this case the cow. and they convert that cellulose into human digestible calories, energy, which is why we're interested in. They also produce several byproducts. One of them is methane, which is the focus of this study. And something to remember is that the methanogens, which produce the methane in the rumen, are mostly archaea in the rumen. There are several options for reducing methane emissions within the cow's rumen. We're going to talk about two methods here, which you can reduce the availability of hydrogen for to combine with carbon dioxide to produce methane gas, or you can reduce the methanogen community present in the rumen. Under normal fermentation conditions, you have high methane output from the cow, you have low hydrogen gas output, and you have high energy output, caloric output for the cow. If you achieve partial methane suppression, you have reduced methane production, but you have very high hydrogen gas production. This hydrogen gas dissolves in the rumen and suppresses all fermentation, and thus you get lower energy output for the cow, so lower productivity. Under successful methane suppression, you get lower methane production, you get a small increase in hydrogen gas production, but that hydrogen gas is expelled by the cow and it doesn't dissolve in the rumen fluid. And then it won't suppress the fermentation, so you get high energy output for the cow and high productivity. So that's the goal. There are many options to achieve this. There are chemical suppressants which can bond to the hydrogen to remove it as a substrate for creating methane. There are antibiotics or vaccines that suppress the methanogen communities in the rumen, or you can use natural nutritional interventions such as seaweeds, which is the focus of this study. There's one seaweed that has been proven to be effective, the red seaweed Asparagopsis, and the sister species, Asparagopsis taxiformes, and another similar species, Asparagopsis armata. They are red seaweed native to Australia and the surrounding waters. They've been shown when added to cows' diets in very small amounts to reduce the methane by quite a bit, upwards of 80%. And this has been shown in the lab and also in live animal feeding trials. The pathway that this acts on, the hypothesized pathway, is a brominated compounds present in the seaweed, which the seaweed produces as an anti-biofouling and antibacterial compound in the seaweed, acts specifically on the archaea methanogen communities inside the rumen fluid. Here are some studies looking at feeding Asparagopsis to cows. This is from Australia. Looks like our label's got a little out of alignment, but they fed Asparagopsis at 0.5, 0.1, and 0.2% of dry weight diet, so very small amounts of the cow's total diet. And over on the left-hand side here, the bars are methane production. And so you see at the high group, methane was reduced significantly. And the line is hydrogen gas production, which has been being expelled from the cow, which has increased significantly. And so this is that successful methane suppression that we were hoping for. On the right-hand side, it's volatile fatty acids, and this just shows that there was no impact on the availability of energy for the cow. Another study from the United States, they fed cows 0.5 and 1% of their daily dry matter intake of Asparagopsis. And again, we had a significant reduction in methane emissions and a significant increase in hydrogen emissions. So this is showing that successful suppression of methane. They also observed no impact on animal energy production or weight gain. So that's great if you live in Australia, but Asparagopsis is not native anywhere in the northern hemisphere, and it's not available on anything approaching a scale that could be implemented to affect the broader livestock emissions globally, or even in Australia. So we were wondering, are there other potential seaweeds that could have similar impacts to the Asparagopsis that might already be available? To answer this question, we conducted in the lab screening of 25 commercially available seaweeds in a simulated rumen environment. These 25 seaweeds covered 11 taxa, species or phylums, and they were from seaweed suppliers focusing in Ireland and Northern Europe, but we also had some from Asia and North America. Our experimental design to test these substrates, we ran 6 in vitro fermentation experiments. We measured 3 response variables, the methane production, the ammonia production, and the pH change. Methane and ammonia are the environmental hazards that we're looking to limit if we're hoping to change the environmental impact of livestock production. And pH indicates a healthy rumen biome, so generally between 5.5 and 7.2 is what we would find inside of a cow. We used grass silage as a negative control. Grass silage is a common animal feed in Northern Ireland, and we also included an Asparagopsis positive control in all of our experiments. We used 8% inclusion rate across all of our seaweeds. This is quite a bit more than the other experiments that I showed before, but we wanted to get a nice clear signal from in our response variables from our test seaweeds. In vitro fermentation is a well-established method for quickly testing animal feed substrates. We collected rumen fluid from 4 individual cows from the abattoir. This was brought direct to the laboratory where it was mixed with a buffer fluid and then added to fermentation jars that contained the ground silage and the ground seaweed. This was all done anaerobically under a continuous flow of carbon dioxide to simulate the anaerobic conditions found in the rumen. We had 4 biological replicates per seaweed per time point, and we took measurements at time 0 and time 24. They were incubated in a fermenter shaker for 24 hours at 39 degrees, and then we took a sample of the head gas for methane. We checked the pH with a probe, and we took a sample of the fluid for ammonia analysis. Here's a picture of one of our ground seaweeds. The seaweeds came from the suppliers dried, and some of them were ground, some of them we ground ourselves. And then these are the fermentation jars that have the rumen fluid and the buffer, and these have been digested after 24 hours with the dried ground grass and the seaweeds inside. In vitro fermentation is a very convenient, cost-effective, controllable, and reproducible way to test feeds in the laboratory. Animal feeding trials are the gold standard, but they're very expensive and very time-consuming. And so we use these results when those aren't possible. However, results from the laboratory experiments like this one don't always agree with live animal feeding trials. And so, but we can use the results from the laboratory to inform or direct future research. It has these benefits and drawbacks. It's also kind of gross. So what did we find from our experiments? We had a large spread of data in our 3 response variables. We found no significant methane or ammonia reduction or pH change when comparing the means of the test seaweeds to their in-experiment controls in a one-way ANOVA. We saw some consistent methane reduction across biological replicates, and when all 4 biological replicates would reduce the methane by a certain amount, but the spread of the data was such that the means overlapped. The highest methane reduction we found was the red seaweed Euchema species, which reduced methane by 19%. And the highest methane production was the brown seaweed Alaria esculenta, which produced 56% more methane than the in-experiment control. Here we'll look at the normalized methane results. So in each experiment, the control here has been set to 0, and the 4 biological replicates have been averaged as their difference from their in-experiment control. So boxes to the left of the dashed line at 0 produced less methane than the control, and boxes to the right produced more methane than the control. Again, the spread of the control data and the test data in each experiment is such that the means overlapped. However, we can see some clear trends that our 4 Alaria substrates that we tested all produced quite a bit more methane than their control. And then halfway down here, we can see this box, which is the Euchema substrate that produced the least amount of methane out of all of our test substrates. Diving into our biological replicates, this is the results, a heat map from one of our experiments. On the y-axis, you have biological replicates, the individual cows, 1, 2, 3, and 4. And then on the bottom, we have our 4 test substrates, and then our Asparagopsis positive control over here. Warm colors indicate production of less methane than the control, and cool colors indicate a production of more methane than the control. So the Asparagopsis positive control is doing a great job of suppressing methane. This Euchema is the seaweed test substrate we found that produced the least amount of methane over here, and so it has a warm color that produced less methane in all of its biological replicates. Same figure for another one of our experiments. Here you can start to see the variability in our data. So warm colors represent less methane production, and cool colors represent more methane production than the control. Here, the biological replicates, the cows, 1, 2, 3, 4, cows 2 and 3, these 2 in the middle, produce more methane in all of the test substrates and more methane than cows 1 and 2. So we had a wide variability between our biological replicates in this case. This Ilaria esculenta here is our highest producing seaweed that we had. So in all 4 biological replicates, it produced more methane than its control. Same results looking at pH data. So boxes to the left of the dashed line at 0 had a lower pH than the control, and boxes on the right had a higher pH on the control. And again, all the spread of the data is such that the means overlapped. However, you can still see some consistent species response to all the Ilaria here. Here we have our ascopylons all grouped together, and so you can see some consistent trends among species. Same results next for the ammonia, and here we had a wide spread of data with a lot of outliers. Again, on the left of the dashed line, these are the seaweeds that produced less ammonia, and on the right of the line are the seaweeds that produced more ammonia. Here we had a lot more variability, and within the control in each experiment as well. So what did we get out of all of that? Other experiments have been run screening seaweeds similar design to this, but no one has published results for the Euchema or the Macrocystis pyrifera species, so these are new to science discoveries for these uses. We have provided evidence to eliminate seaweeds from future studies, such as our Ilaria species that produced much more methane, and these don't need to be studied further in this way. And we found no effect on ammonia production or pH, and so if other benefits are found, these seaweeds can be fed to cows without affecting these outcomes. In terms of what's next, we've only begun to scratch the surface of available seaweeds for this potential use, so we can screen more seaweeds in this way. Potential high performers from those studies could be led into animal feeding trials in the future to corroborate the laboratory results. My abstract was accepted at the European Federation of Animal Sciences Conference in Lyon, and we plan to publish this chapter of the PhD project as a standalone paper once all the results are in. So thank you very much for your attention.
Thanks. Really interesting, important subject.
Thank you.
You only have one really good species here, yeah? Or was
Yes,
it the one here?
that was the highest performing that we saw.
Yeah. So how would you like to go further with that species? Well, you said you were going to test a lot of other ones. Why not go keep focus on one?
The next step for if we pick the Euchema as a high performing one would be to test it at lower doses to see if it had similar effects. We chose 8%, which is to give us a nice clear signal in the response variables, but that's approaching a safe limit where you could feed a cow where salts and certain minerals become unsafe for the animal. And so we'd have to test it at lower concentrations to see if it still had that effect.
So today, how much do you get?
That was one of our commercially available seaweeds. So I think I don't know if you can get enough to feed to a large cattle farm, but you could feed it to an experimental herd, certainly.
That's the real problem. To scale it up.
Yes, that was our hope.
It's a lot of cows.
Yeah, our hope was to find a commercially available seaweed that would work as is, but it appears that they're not as effective.
Thank you for the presentation. Scientific research has basic climate change and circular economy and things like that. I have a couple of questions. What was the rationale for the initial selection of seaweed? It's a mixture of species that are native for the Atlantic, while you have some others like Macrocystis, which are not present in the Northeast.
Yes, the selection process was picking seaweeds that were available at large scales, that could be ready to be implemented for use in large scale animal livestock operations. There was an eye towards Irish and Northern European producers, but in order to cast a wide net, we picked North American and Asian producers as well. So the Asparagopsis is not native to the Northern Hemisphere, and so we could in theory grow it up here, but that is not viable because it's not native. But if we found a seaweed that was already available in large enough quantities, we wouldn't have to worry about growing it. So that was the.
And the question is, the media problem with translating the results from the in vitro experiments into experiments regarding the presence of other secondary parameters can be toxic or can affect the system process or something like that. Have you done any screening of whether those we have some other compounds can be actually problematic in terms of decision?
That's certainly a next step. Analysis that we didn't get to was analyzing the volatile fatty acids as they're produced in the fluid over the 24 hours, and in the ratios of the various compounds that are produced in the digestion, you can see if digestion is going along as you'd expect it to in the cow, or if it's diverging in some way. And then if it's diverging, you need to look back and see what's happening. So this is very much a preliminary screening to see if any promising. But it's very true, if you're gonna provide this to animals, there are many other assays you need to do. And we did some others. We looked at the fiber content of the seaweeds that we had, and we looked at the phenol content, and also at the oxygen scavenging capacity. And we put them through a broad spectrum mass spec to look at the volatile compounds, the organic compounds that are in there. But all of that needs to be parsed out very carefully before it's ready to be fed to animals in any kind of way.
Thanks for this strange orientation. That's interesting. So I'm curious that do you have an idea which is the chemical component in Asparagopsis reducing the production of acids?
The hypothesized compound is bromoform, and the red seaweeds produce a lot of halogenated compounds that they use for various means. A lot of them are antibacterial in function, though if that's what the seaweeds use them for is not always known. We did, as we put all the seaweeds through the mass spec, looking at a broad sweep of volatile organic compounds, and the data has not been nearly parsed out as thoroughly as needs to be before it's presented anyway, but the Asparagopsis has 3 or 4 times more halogenated or brominated compounds than the next seaweed in line. And so it is a very complex seaweed with a lot of organic compounds that could work.
I presume that's not practical to use seaweeds in the cows, instead it has some chemical that
Yes, there are. There's people have used chloroform as an option, and there's something called 3-nitro. It's called 3-nop, but it's commercially available and farmers can buy it. But it leads to lowered production because you get the dissolved hydrogen gas in the rumen, and so it does suppress methane production, but it also suppresses energy production for the cow. And it hasn't been proven to be as effective. You get 40% reduction, whereas the Asparagopsis you can get 80 or more % reduction. Yeah, I have here is the same methane production, but with our positive control included. And so this is the Asparagopsis positive control, which is just by far the most reduction that we observed.
Thanks. Thank you. I have some questions about the variability between cows.
Okay, sure.
Yeah, because that's the source of the issue. That's a big issue.
Yes.
So how consistent was it? Was there a cow that was particularly resistant to being amenable?
These 2 cows, numbers 2 and 3, in this experiment showed, so again, we had 4 biological replicates over 6 experiments, so we had 24 cows in total. And we unfortunately did not know anything about their life history or their diets before that. They were all from Northern Irish farms, which have a very consistent grass pasture or silage feeding regime, but we're not sure if we know that they were healthy, we know that they had healthy rumen, but we're not sure when they were last fed or what they were last fed, and so that can have a big impact. Another technique is to homogenize the replicates together. So rather than biological replicates, you get technical replicates. We opted to keep the cows separate so that we could get a higher resolution in our data. However, that led to a higher variability in our data as well.
So I was wondering about those records when you took the 2 time periods. Did you take down one normal and put the 1 time period?
Yes, we had 8 for each, and so we mixed them all together randomly, and then we put the 24-hour ones in the fermenter, and we put 0-hour ones right away, and then got rid of it.
So, how much work is going on in the seaweed production line to try and, to manufacture this, to come up with a simulated, 40 years enough bioinformatics work to try and produce this stuff?
To try to grow a potential seaweed at a large enough scale?
Just produce the compound, the active compounds.
There's a lot of interest, there's a lot of money in agriculture, and with the incentives to reduce carbon footprints of various sectors, there's a lot of money going into reducing kind of the low-hanging fruit. And methane is 20 times more potent than CO2, and it only lasts for about 30 years in the atmosphere. And so if we can suppress methane now, we can have huge payoffs in the future. And so this is considered you get a lot of bang for your buck by suppressing methane. So there's a lot of money going into trying to look at that. How?
What is the progress?
I don't know. Australia is harvesting this from the wild, and there are some companies that are trying to grow it in mass, but Australia has a huge livestock agriculture industry, and so even just feeding that one nations with its native seaweed will be immensely difficult. But I know that they have some legislation saying that farmers have to do more to reduce their methane, and so they can reduce their herds, which is the surest method by far to reduce their methane. You can use the chemical additives, or you can try for natural.
And one last question.
Sure.
How's that seaweed?
At these low percentages, they've been shown to not have a bias. At higher percentages, they have been shown to not like it as much.
That's your upper limit.
Yeah.
Are there any questions from? I was wondering, all those are you using the traps or
we used the entire, I can go back a little bit. We used the entire ground seaweed like this. Some of them were oven dried, some of them were freeze dried, but it was the whole biomass was ground up. Some studies have soaked it in an oil, which is apparently easier to feed to cows, and they've shown that that's still effective. So there are other pathways you could use, but we used whole ground. Try it.
I was wondering, what are the
Sorry? Wow, it is this, it's called Van Schoest solution, and it is supposed to simulate the saliva, which is in great quantities inside the rumen. And it is also a pH buffer, so to normalize the pH between the rumens, as long as they're in a range. And also has an oxygen indicator, so if oxygen is present, it turns pink. And so we know that we can't use it if it's turned pink, so we need more CO2 to bubble through it. Thanks.
We have a little bit more about why you chose that was a regional thing, also like why.
We did as many as we could in the time. We actually ran about 9 in vitro experiments, but we had to redo a couple of them because one of them we had to redo, we had anomalous results. And there's one that the results are still pending, the methane results, which we sent away, are still pending. So we had a big list, and we kept adding more to the bottom of the list, and then we did as many as we could working through.
Thank you very much.
Okay, thank you.
click the presenter there and deactivate and activate again.
You activate it again? Okay, perfect. Do I have to Huh? What, do I have to do something? No, it's okay. Yeah, we had a problem with the streaming. Yeah. We'll be fine. Okay, so here. Okay. Is it fine? Can I go?
So good afternoon, everybody. My name is Gianile Benetti. I did my thesis at the Biological Center for Marine Science of the University of São Paulo, and my supervisor was Hudson Terço Pineiro. And my thesis is about the high differences in the littoral formation between rocky shores and reefs in southeastern Brazil. So, to introduce my topic, I'm going to start talking to you about the anthropogenic marine litter, which nowadays is the main threat to the marine ecosystem because it's ubiquitous to all the marine realms and is affecting marine life in many ways. Like, for example, many animals can ingest litter or get entangled into it, or just because the plastic and the other substances release toxic components into the water. Also, in the 2030 Agenda for Sustainable Development, plastic is indicated as a key indicator for marine pollution. So it's very important to tackle this problem and to understand why and how plastic is there and how it's affecting marine ecosystem. At the moment, most of the study, actually more than 95% of the study regarding marine litter are focused on sandy beaches and coral reefs, just because these environments are more easily accessible from the public and the scientists, and because they have a high economical value due to tourism. But this led to an overlook of other very important environments, such as, for example, rocky reef and rocky shores, that are still very important because they provide with coastal protection, and actually they are also hotspot of biodiversity, and they also host many important fish species that are important for fishery and commerce. Furthermore, most of the study nowadays focus only about the composition of marine litter rather than why the marine litter is there, so the driver of litter accumulation. For all these reasons, the aim of my study were to identify the main driver of litter distribution and accumulation on rocky shore and reefs, but also to compare the composition and the types of litter in these two environments to see if and why there are differences among them. So this is the study area where I sample my data, and as you can see, is in the littoral area of the state of São Paulo in Brazil. I sampled 11 sites, 4 of them on the mainland, 4 of them on the coastal island of Ilhabela, and 3 within the marine protected area and wildlife refuge of the archipelago of Alcatrazes. The sites from 1 to 11 reflect a gradient of distance from the city center, but also they have different status of protection. Like, as I said, the archipelago of Alcatrazes is a marine protected area, but there are other sites like the number 4 and the number 5 that are actually they have some kind of protection. Like, for example, is not allowed to fish, not even for recreational fishing. To sample, I did bed transit, both on the shore and in the reef, but to sample the reef, of course, I dived using the underwater visual census technique, which basically it means that for both the environment, I dived or walked on a line of 20 meters, looking at my right and at my left for a distance of 1 meter, counting the trash that I encountered. It's very important to note that all the trash was counted but not collected, unfortunately, and that only the macroplastic and macro litter was considered, so everything bigger than 5 millimeter. At the beginning, all the trash was divided into 2 main materials, which is plastic material and non-plastic materials. And furthermore, then 5 categories were used for each of these 2 main categories. For the data analysis, it's important to say that I did 256 transects, an average of 6 transects for the shore and an average of 18 transects per site for the reef. I know the numbers are different, but that's why for the statistical analysis I standardized the number of pieces of litter encountered per number divided by the number of transects per site. So the data were standardized and it made actually sense to compare them. And to compare the composition of trash, I use principal component analysis and principal coordinate analysis, while to investigate the driver of marine population, I use generalized linear model. And in particular, I consider 6 different factors, which are distance from the nearest beach, distance from the nearest town, distance from the nearest harbor, the number of people living nearby in an area of 20 kilometers radius, the wave energy, and the current velocity. I am going to start illustrating the trash composition. So the first difference among the two environments were the abundance of trash, because as you can see on the shore, the number of pieces of litter per transit was way higher. And this is probably because on the shore, you have to take into consideration that the litter reached there because transported from the water, but also because direct dumping or the wind. And also, I will show later that also the composition is different. So there are type of litter that can go farther away due to floatability. Also, the distribution of trash was different because if we consider the site, for sure, the site where the higher amount of litter was encountered was within the MPA, and this is again because of the floatability and the type of plastic litter that we encountered, but also it's due to morphological shape of the site. In fact, for example, site 9, which is the one where the higher amount of litter was encountered, is a bay that is directly in front of the direction of the current, so is actually retaining a lot of the litter. While for the reef, the sites with the higher amount of litter were the one close to city and to beaches. In the graph to the right, you can see that in blue there are the reef sites, and in green there are the shore sites, and you can see that the composition is different. In fact, these two sites are the two environments are actually separated. Talking about, in fact, the composition, plastic was the main materials, once again, because it's always like this. And in the bar chart, you can see in the left the reef and in the right the shore. And we can see that the composition of plastic is quite different. In fact, for reef, the most abundant type of trash was related to fishery activities, while on the shore, the most abundant type was polystyrene, and this is what I was talking about before. So polystyrene is very floatable, so it doesn't sink, and that means that the current can actually transport it farther away and get entangled rather on the shore than on reef because it doesn't sink. And also packaging plastic was rather important, and in fact in this graph you can see that packaging plastic PA and polystyrene PO are the two type of plastic that are more responsible for the separation of the two environments. Talking about the sources of anthropogenic debris, these bars represent shore sites, and the last bar represents the total. And we can see that there are many differences. For example, I categorize the trash in 4 categories, which is in yellow you can see local trash, so basically direct dumped by tourists or people working nearby. In gray, there is the trash related to fishery activities. In blue, there is the one coming transported by the water, by the sea. And in green, there is the one that was not possible to classify. So in some sites, the trash that was not possible to classify is a lot, as you can see. And this is because, like for example, if there is a plastic bottle in a bad state on the shore, we cannot say if the plastic bottle was left there by a fisherman or was transported by the water, so it was not actually possible to define. But the important thing that we can see from this figure is that there is a high variability among sites in the same environment, which means it's very hard to tackle this problem because when we want to find solution or to propose regulation, we have to actually go very in deep and very local area because as you can see, for example, The site 5 and the site 6 represent shores that are very close to very crowded beaches. And you can see that in this case, main uh, the main source was local because people just dumped trash, and also because these sites were close to construction site, and we noticed that the constructors were just throwing away stuff that they didn't need anymore. While, for example, in the MPA, so in site 9, 10, and 11, all the trash is basically transported by the water, and the few fishery-related items are because of illegal fishing inside. So we can see that already there are different problems because different sites require different solutions. Especially, for example, I think it's very important to talk about fishery-related trash because Brazil has a very important culture of fishery and boating. And in fact, we can see that, for example, for shore, only 14% of the total trash, in fact, only 62 items out of 421 were related to fishery, while in the shore, basically half of the trash was related to fishery, which means that it's very important to try to talk with the fishermen to explain to them that they're actually damaging their own source of money because leaving behind ghost nets and stuff like that, they are gonna impact the status of the stocks. Lastly, talking about the drivers of trash distribution. In green there is shore and in blue there is reef. My GLM identified two drivers for each of the environment, which are the number of people living nearby was significantly important for both the environment, while for the reef was very important the distance from the nearest beach, and for the shore the current velocity. So on the x-axis we have got the current velocity or the distance on the number of people and in brackets the site, while on the x-axis there is the average number of pieces of litter per transit. And so we can see many things. For example, the Red Cross is because in that site was not possible to collect data for shore, so we only have data for reef for one site. So, for example, we can see in the first bar chart that for shore and for reef, the number of people was important, but has two opposite effects. In fact, for shore, the number of people, when the number of people increased, the number of litter pieces decreased, and that makes sense because, as I said before, we encounter the higher amount of trash in the MPA, which is far from the city center. While for reef, with the increasing of the number of people, also the number of litter pieces increased. And this again makes sense because before I said that, in fact, we encountered the higher amount of litter close to crowded beaches, which were more close to the city center. And the same thing it's valid for the distance from the nearest beach, where you can see that when the distance increase, the average number of litter pieces decrease. So to conclude, I think, it was kind of obvious, but of course you always have to check. And in fact, also in this study, plastic was the most abundant type of litter, being more than 80% in both of the environments. So, as other study about Brazil already suggested, is one solution could be for the sites close to city center and beaches to increase the number of bins, because I also were there and there are not many bins. So I think can also be a problem why people just leave it there because they don't know where to put it. And also it's very important to educate citizens, but also fishermen, because they don't really know which is the actual impact that their trash is doing on the environment. It's also very important to pay attention to daily habits and to disposable plastic solution for two main reasons. First, because one of the biggest categories of trash that we found was polystyrene and disposable plastic, packaging plastic. And this is bad because people are we should teach them or make programs to teach them to buy more usable plastic, and the same problem is with polystyrene, because polystyrene is not recyclable, it's not reusable, but we are keeping producing it, and in fact, many of the shore sites were highly impacted by pieces of polystyrene that within the other things keep getting smaller, which then can lead to bioaccumulation in fish and marine life that we also end up with. For the future, I think it's very important to agree on a unified protocol to study marine plastic, because when I was reading other articles to learn what to do or to just have an idea, each article had a different protocol and they were considering marine litter in different ways, counting different things, and it was very hard to just have an idea of what is the status of marine pollution in other areas of the world, just because everyone is just doing something. So I think one of the main goals would be to have a unified protocols and also to investigate more this overlooked environment. Thank you.
Sorry. Okay. Thank you. When it comes to sources of the litter, you had the number of stations with 20 kilometers.
Here?
Yeah. That's probably quite well. My experience from Swedish restaurants. But anyway, is that much of the litter comes from further away, and much of the litter can be traced to a number of other countries.
Yes.
So I just wondered, is there any large scale currents or things like that that you use as well?
Yeah, so there are, yes. So regarding this, there are two main answers that I can give, because there are some studies that actually even evidentiate this. So when we look at this study, we should say, okay, it's very important to make study on a larger scale to really understand the currents and why the trash is coming. And this is true for sure. But there are other studies, like for example, mine already, that are showing that within a small area, like the one I surveyed, it was 40 kilometers, so it was a very small area. There is already such a huge variability among sites. So I think it really depends what's the goal of the study. But I also think for sure we should study on a global scale this problem. But at the same time, we should to really solve the problem on a local scale to actually make a difference immediate, fast. We should also do small scale study because of these reasons.
I agree. But also you have this high variability because you have like could have like a bay that
yes, it's really
the sea the bottom.
Yeah, like sideline.
So that's that you have this.
Yeah, no, for sure. I think there are. I have a shorter time to do this study. I only have 4 months and a half. So I definitely think it's way more complicated than this, and there are way more factors to take into consideration, like already the morphology of the sites, the velocity of the current, the direction of the current, where the sites are co-located current. So for sure there are many more factors and analysis to take into consideration.
Yeah, thank you. I have one question regarding, can you go back to the slide? I think it's the previous one. The other one. How did you sort the resources?
Okay, so to decide to divide my litter in these 4 categories, I actually based myself on a study of ports et al. From 2011, where they were doing something very similar, and they decided to divide the trash in these categories. And I thought it made sense for me as well, and so I followed that study. Like as I said, there are many different protocols, and for this specific thing, I choose to follow them, but there are so many more ways. Just a piece, I consider it unknown. I consider when it wasn't not possible to know for sure, I put all in the unknown. So I only choose a source of the other 3 when I was sure, when it was actually possible to categorize. When it was not possible for many reasons, too small, no way to understand, I put in unknown.
So sure that there is incoming to site 4 was coming from the sea.
Okay, so for example, because like there are many small pieces, for example, of polystyrene. So I took into consideration many things, like even, for example, the position of the site. Let's say when it was into the MPA, I'm sure it's coming from the sea because it's very far. You can only go there from a boat and you cannot enter. So there are no people living on the island. There is no way that type of trash is coming from local sources. For other sites, like for example 8 and 7, it was a little bit more complicated because they are remote sites, but at the same time, there are some fishermen going there. So, for example, if there was a shoes, it was coming from the sea because there are no people living nearby. But if there was a bottle and it was kind of good conserved, for sure, 2 days before, a fisherman was there and leave the bottle of water. So, of course, it's a personal interpretation, but I did my best taking into consideration many factors.
Why is site one missing?
Because as see, I should have said here as well, but as I said here, wait, here, for site 1, the data regarding shore are missing because it was like a kind of a small island in the middle of the sea, but it was not really a island, like there was no shore to walk on. It was just rocks, but it was not possible to walk on it, so I couldn't.
Okay. Yeah, and so you can see there is large variability in transmitted and did you consider it has more factorial sampling design and days and also localities in two
Like to divide base and
or island and mainland,
no.
remote places and the closed cities
Okay.
because results are ID. So you can identify which is the factor
Yeah.
responsible. Did you consider that?
No, I did not. But I think this is a very good advice, and I think I would try to do it, and maybe the result could change a little bit. But for now, we only consider the site separately, so we did not like classify them. One thing that I did that is in the appendix is to do a clusterization of the sites based on, but this is based on trash composition. So the cluster are based on the composition of trash. And here is possible to notice that for both for reef and shore, the tree cluster does not reflect the mainland, the coastal island, and the offshore island. This division is not respected.
No,
so
I am not saying that.
they should, yeah. But in
easier.
Mhm.
So at least in preliminary something science. I can also related to the question of the more people coming far away and perhaps considering remote places, very remote, in which only long distance
trash would come.
can arrive, can be also
Yeah.
control baseline, know the amount of people is voting.
Yes. So this could be a further like step more to do.
Yeah.
But if we consider the study area, and you can see it here, there is like this small one, the small island, and this is the main island. Actually, the channel between the mainland and the island is there is a strong current into it, and the current goes directly into the offshore island. So, to be fair, it's highly probable that the trash on the offshore island is coming from South Sebastião and the island because of the shape of the channel and the current. So, it's not super likely that trash from very far is coming in that location.
Yeah. And the factors, so you classify the you see different environmental factors.
Yeah, these factors.
But they are responding already.
Yes. Yes. And in
Do you
fact,
think that's a problem?
yes, it was. In fact, when I ran my generalized linear models at the beginning, the first time I ran it with all the factors together, and the generalized variance inflection, the VIF was super high, was higher than 3000. So actually then to run the best model, I removed many of them because they were highly complete. Yes.
Thank you very much.
Thank you. Thank you.
I guess I have one question about GLL. So,
Yes.
so what's your best model?
The model? The best model?
Yeah.
So, you want to know how I arrived to the best model or what?
No, no, what it was.
Okay, so at the end of the day, after that I removed the factor that was strongly correlated, I actually remain with two, in one case with two factors that are the two that actually are the best, the more significant.
So you have three there.
No, sorry, because there are two for the shore and two for the reef. But since the number of people was correlated to both environment, I put in the same graph. But I had two, like you can see here, sorry. You can see in this tab that for shore I had two factors and for reef I had two factors. But actually, in one case, I think for shore, I already only have two factors because all the others were strongly correlated. So I think the models were not super strong because of this. So I think I should consider more diverse factors.
Is that why you present the results in a GLM? And the outline?
Okay, the main reason why I presented my GLM like that is because I have not a super strong background in statistics. I'm more usually I work with genetics, so this is very far also from my field, but I tried, and this is the very first time I put myself into statistics, and this is the best I could do in a short time. I tried.
It is as illustrated. The results. Are there any issues?
Two- 256 transects, but together shore and reef, and just because I didn't dive alone, like I had other divers helping, and actually when we went to the offshore island of Alcatrazes was a bigger expedition, so there were many divers from many universities that were involved in another bigger programs, and they helped me also since they were already diving, counting like benthic organism. They actually also count the trash in transect for me, so I received help to do all these transects. It's too much. Yeah.
If you figure categorization of concept,
Yes.
you do it based on your eyes. Like you make your own categorization, all these categorization algorithms. I know there's no like
Yeah. Like if you see my raw data, like I did all the for each item I did the categorization. But basically, yes, I did it by hand. Like when I know something is polystyrene, you can see is polystyrene, and not mono-use plastic. Like I consider items that are clearly not disposable. Like I found shoes, or I found razors, or I found toothbrush that are made of plastic but are not clearly not disposable items. When I couldn't know because the piece was too small or too ruined, I put in others. Fishery-related plastic, I count nets, ropes, and fishing lines that are clearly related to fishery. And for packaging plastics, all the stuff that you can reconduct to packaging. And when I couldn't know, I put into others. Because we didn't have a lab, and I actually also didn't have time to do chemical analysis. So that's why I did these big categories, not really into the type, like the chemical type of plastic. I work with the macroplastic, but there are many studies focusing on microplastic, but again, especially on beaches and coral reef. Like there are very few studies on microplastic and rocky environments. It's something that we should do. Thank you.
It's okay. Yeah, just not sure. I'll just see. Okay.
Okay, hi everyone. My name is Skylar Reese, and I want to start with a quick question for the audience, which is, what do you think this is? Quickly, anyone? Salt, one more. Ice, microplastic. This is a biofilm, so a composition of living organisms, bacteria, diatoms, that will form on the surface of plastic when it gets polluted in aquatic and marine environments. That brings me to my thesis topic, which was understanding biofilm assemblages on aquatic plastics and their potential as a dating tool for plastic pollution. This was done in collaboration with the University of Cambridge and my supervisor, David, and funded by the Erasmus scheme. Now, as Jaeli just said, plastic pollution is a huge issue in our environment with a ton of negative impacts. This is quite well known at this point. What is a little bit less well known is that this plastic pollution will harbor unique microbial assemblages, like I showed you in the first photo, of bacteria and diatoms, forming its biofilm. Now, this biofilm will impact the local ecology where the plastic has been polluted in, and also impact the plastic itself. So let's just start with ecology first. Right, this plastic is full of living things. It's going to impact the nutrient cycles in the area, especially micronutrient cycles. It can act as a vessel for pathogens. Things like Vibrio bacteria can live in a biofilm and then get introduced to the environment. There's also a food source because it's a bunch of living things. And then finally, it can attract biofouling organisms that will come and sit on top of this plastic. And some of these biofouling organisms might be things like invasive species, like zebra mussels or something. Now, the biofilm will actually impact the plastic itself. I'll just hold this as an example, right? So it's a film on top of the plastic, and it will change its density if the biofilm is so great. What might have been a floating plastic can have the biofilm on top of it, and that can become now a benthic plastic. In some cases, too, you'll get this biofilm, and it will actually prevent the UV from coming in and causing photodegradation, so it kind of preserves the plastic in the environment. On the total other hand, though, bacteria can live in this biofilm that are actually capable of doing plastic breakdown themselves. So it really depends on the biofilm composition. But for this thesis, we looked at something completely new, which is, can you use biofilm to inform how long a plastic has been incubated in an environment? So saying, how long has plastic pollution been there just by looking at its biofilm? This is a big question mark. So first, why even date plastic? Well, we've been dating plastic since plastic pollution has been an issue. It helps with environmental impact assessments. You want to know what plastic types are taking the longest to degrade, where are they taking the longest to degrade. You also want to target your cleanup efforts. You want to target these towards places where plastic pollution is a very long-standing, pervasive issue. And actually for this project, I also worked with Thames 21, a small nonprofit group looking at UK waterways, and they just acquired all these active plastic collectors to help them clean up UK rivers. And one of the factors they want to consider, or where they should put these active litter collectors, is where plastic pollution is sitting the longest. And then finally, just for legislation and public awareness, to really know how long plastic is living in our environment. But like I said, we've been dating plastic since plastic pollution has been a problem. It's just that these methods can be quite expensive or they can be quite technical, which are up here. So something like FTIR, which is a really good method. A group like Thames 21, the nonprofit, has no access to such machinery, and they were quite interested in what we proposed, that the biofilm on the plastic may offer the simple plastic dating tool. So why did we think this? In 1989, a man named Martin Wall came up with a marine epibiosis model that basically said biofilm is going to accumulate over time on a substrate. Doesn't have to be plastic, any substrate, when it is in an aquatic environment. So you start with the incubation. You can imagine that's a little plastic. 1 minute, you're going to get some macromolecules. An hour, some bacteria. a day, some diatoms, a week, some larvae, a month, some algae, and then from there on out, some larger biofilms. While it might not be interesting to date a plastic to the hour, no one is trying to do that, what we really want to take away from this model is that biofilm is going to increase over time. Now, like I said, this came out in 1989. There's been great work on biofilm since then that have introduced some nuances. The two nuances I was interested in is that different plastic types will harbor different amounts of biofilm. You can think of something like styrofoam with all of its nooks and crannies and lots of surface area will have more biofilm than something that has like a smooth surface. And then also site and hydrological conditions can lead to different biofilm abundances as well. So taking all of this into consideration, we proposed our 3 hypotheses. Our main one is that biofilm abundance will increase over time on our plastics, but also taking into consideration that plastic types will host different biofilms and that site conditions, including salinity, maybe some hydrological conditions, will influence the amount and composition of biofilm as well. So how did we test this? So we set up a in-river experiment in the Great Ouse in the UK. I took 3 common plastic types. We have polypropylene. This is kind of like a wet wipe material. We have polyethylene terephthalate. So this is like a water bottle. And then polystyrene, styrofoam. Cut them into 2 by 2 centimeter coupons, strung them on hemp lines. This is what you're looking at in the bottom left. And then put them on bamboo stakes. Took these in triplicate, so 3 bamboo stakes went to each site. This is our map here. Site 1 is most freshwater, site 2 is estuarine, site 3 is marine / estuarine. This is the bay here. And then for 3 weeks, we would go and we would take each 30 of each plastic type, bring it back to the lab. So there I am, getting the plastics. We would then air dry them in a sterile petri dish. At this moment too, we would take photos of the plastics. So this is our site 1, our site 2, our site 3 over time. Now we would stain them with 0.1% crystal violet. So quite simply, crystal violet is a gram-negative bacterial diatom stain. So it's going to stain our biofilm on the plastic. Our plastics will absorb this purple stain. Then we will go through an elution phase with ethanol. So we'll put the plastics in ethanol. The biofilm will then release all the crystal violet. So we just have ethanol that has now been dyed purple. We would put those in an Eppendorf tube, transfer the liquid to a microplate. So you can see that on the left here, and then read the absorbances with a microplate reader. So very, very, very simply, the more purple, the more biofilm, because it was the more absorption. And then we didn't stain all of our plastics, and we were interested in the composition as well. So we took some SEM images, like I showed you at the beginning, to look at that. So what did we find? We were, at this point, I had a huge Excel sheet with all of these absorbances for all these different categories. The first thing we did, we ran a permutational analysis of variance to see how absorbance was affected by time, site, plastic type, and the interactions between these guys. We found that all of our terms were significant except for one interaction term, but we found that site was the most determinant factor for absorbance. We can look at this a little bit better here in this graph. So I'll just orient you guys. This is our control in the top left. Site 1 is our freshwater, 2 is estuarine, 3 is marine. On the x-axis, you have the 3 plastic types. On the y, you have absorbances, and then the colors are the weeks. Controls, you don't really see anything, as we would have thought. Now, site 1, we see something very interesting, right? We thought that biofilm absorbance was going to increase over time, like this model had told us. We would see a linear trend here. Actually, the green are week 2. There's a huge peak, and then it drops off at week 3. But now when we look closer at site 2 and site 3, you see a little bit more of what we were expecting to see. General increases in biofilm over time. So what is happening at site 1? What is happening at this freshwater site? We looked into the few and far between literature on biofilms on freshwater plastics, and we found that the hydrological conditions can be quite important in determining how much biofilm a plastic will have at any time. We got some hydrological data ourselves for the River Great Ooze. Again, rainfall on the left, river level on the right. And we're really interested in between these bars of 9.2 and 3, because this is where we see the weird drop off. What we see is a huge peak in rainfall, a huge peak in river level, also a peak in river flow. So we think that there might have been some sort of, physical washing of the biofilm off the plastic. The salinity might have also changed with this rainfall event. The nutrient composition of the water, things like that. But the big takeaway here was that although the hydrological conditions were similar across all sites, it was the variable nature in these riverine habitats, not as much in the estuary or in the marine, that really complicated the biofilm development. Kind of got away from the model that we thought we would see. We also wanted to see who was living there, not just how much. So we took some SEM images. This was after the 3 weeks, so this is just one time point. The top, we have our sites. The left, we have our plastic types. We can just start with the top row. Top row is a polypropylene wet wipe. Site 3, you're seeing the strands of the wet wipe. You're not seeing any biofilm, diatoms, things like that. But look at site 1. It's completely covered. You can't see the strands at all. And site 2 is kind of a bit of an in-between. PET was quite smooth. What you're looking at here is mostly just the plastic surface. And then polystyrene was a bit of an in-between. But now the takeaway here is that although we saw this kind of strange event in peak in biofilm and then going back down, still at week 3, these riverine plastics at site 1 harbored the most biofilm, even despite this variability. We did a kind of quick and dirty community analysis of the diatoms. We could not get to species or genus or anything. We followed a characterization that had been put out by a guy named Carson in 2013 of just diatom classes that we could actually see from the SEM images. And we wanted to see, are there similar trends by site, by plastic type, as has been proposed in the literature? And you can kind of see here in this plot already. There were no singular trends in diatom proportions. You can't just look at site and say you're going to see this much proportion of diatom. Same with plastic. It's really this interaction between site and plastic that complicated our model. We went on to just kind of confirm this statement with an NMDS. On the left, we're looking at plastic. So our 3 plastic types are the 3 different colors. Same here for the site. We're not seeing significant communities in how we characterize the diatoms from this NMDS. And now moving on to our discussion. So original hypotheses, just going back to that. The first one, right, we thought biofilm abundance will increase over time. These are the models that were in the literature that we thought we were going to see, be able to date the plastic. But then we saw site, not time, was actually the most determinant factor of the biofilm abundance. This is in agreement with some studies, but again, not all. Most studies will say it's time or it's plastic type, but we were one of the first to actually look across a river, across a salinity gradient, which brings me to my second hypothesis. Site conditions, including salinity and other fluctuating hydrological conditions, are going to influence the amount and composition of the microbial communities. And like we said, site was super important, the most important. The salinity gradient determined the biofilm of the plastics, or at least the abundance of the biofilm of the plastics. And there's another great paper out there that kind of found similar things to this. Finally, we thought plastic types will host different microbial communities or different amounts of biofilm. We really didn't generally see this. We saw that the polystyrene wipes hosted the most at some sites, while other types of plastic hosted the most at other sites, so it was a little bit variable. But big takeaway, no plastic-specific or site-specific communities. I want to talk about our limitations. Of course, we were limited by time and resources. Unfortunately, we had to put our plastics in formalin to save them for the SEM. which prevented us from doing molecular techniques in the future, which is quite sad. There's great papers out there that say, there's specific assemblages if you can get down to the species level. We were not able to do this. We were also unable to get data on the nutrient levels, these things that I thought would be super interesting during the hydrological event. Finally, our big takeaway conclusions, what we think we contributed to the literature. Biofilm abundance did not follow this typical additive epibiosis model, possibly due to hydrological conditions. Second, the interaction between site and plastic type really complicates this biofilm formation. There's no simple model out there. But we do think if a model is ever to be formed, which TAMS 21 is still quite interested in, we think we might have given a starting point to this and ID'd some key inputs to this model, such as you really need to be careful about hydrological conditions, and it's probably not super feasible to make a model over a whole river, and it might not have to be very site specific. So thank you everybody that helped me out, my lab in Cambridge and everyone, and yeah. I'm scared.
Thank you very much for the first topic, plastic management. And I wonder if you think that's a good method to evaluate the plastic age. Perhaps in the process, what about plastic traveling around the world and different environments, different communities, and different how do you age?
So I think this work is great evidence that it does not work at a really, really large scale like that. There's too many variables. But we still don't have a good dating method to this day that is both accessible, cheap, doesn't require a lot of technology. So I think, I do still believe in giving this a shot in small, kind of contained environments like one estuary or one river system, which a lot of these, at least in my experience over the thesis, a lot of the small water nonprofit groups we were working with kind of just focus on, a small bay or a small marsh or something like that. So in that case, probably.
And so for me, that's probably not simple to age. And do you really think it's relevant? No pH the plastic is floating around?
Yeah.
That's my question.
Yeah, no, totally.
Why bother?
Yeah, that's why I made
It's plastic.
this slide.
Yeah,
why
yeah,
date plastic? Cares to even date plastic? But without small nonprofit groups having access to this data, again, it's harder to maybe target their cleanup sites. They have information from currents of where the plastic is ending up and things, but from the people I spoke to, they're actually quite interested in where plastic has just been sitting for a long time, and they don't know that just by field sites visits over a few months or things like that. It goes over years and years.
Yeah, thank you. It was already a presentation. I have one question. I want to focus only on environmental factors like salinity and things. But at the end, the process is a process of polarization. It's available space, plastic, and things colonize there. Could it be that the different, for instance, diet composition in different parts of the river actually compromise the community that gets attached to the plastic?
Absolutely. So there's different literature out there, but the general agreement is that the microbes living in the water body are also going to be a very significant factor in who is going to live on the plastic, but not necessarily in terms of abundance, or at least not in the papers I've seen. But again, you need to get down to the molecular level. You need to be able to do molecular techniques to find this. I would have to take eDNA from the water to figure out, the microbes in the water and then compare with the plastics, which is something, if I had more time and money, would have been super interesting, and that's what other papers do.
And then, but on part of the studies, we showed abundance of particles, stain But it maybe the case also that when diatoms, because we assume that there is a linear process of colonization.
Yeah.
But maybe it's not bacteria, then diatoms, and then macrophages or whatever. Maybe when the diatoms make a new community, they also favor some bacteria.
Definitely. And this is something my supervisor brought up too. Or even it's the fact that when you get that diatom phase, maybe you're attracting more kind of predators or feeders to come and to get rid of the biofilm from the plastic. So it's much more variable than the original
one. Okay, thanks for what about the age test?
Yeah.
But if you want to do that, still, you showed some pictures in the beginning with the images that must have been, well, plastic that been much longer time than 30 days.
These.
Yeah, well, you know,
this.
Doesn't matter. But where you also have long things like that. Do you relation in time? 30 days.
Definitely.
Yeah. So this was more like a method. For how long would the ones that you want to have the data? Is it a G or is it what is it?
So we have this wall model that says after 30 days, the actual biofilm, so not huge biofouler's, but the thing that's actually like whatever, stuck to the plastic, right? It will become stable after 30 days. Literature out there says that. Other literature says 6 months. So in a perfect world, and what my supervisor would like to do in the future is look over the 6 months to actually see the variability after that, because it could look totally not linear, totally different over time.
Yeah, I mean, you must come up with some climax community after the ones that stabilize something that's a little bit more stable than initial.
Definitely. And so when we were in the river in the Great Goose, we did find plastic that we didn't put in the river, and it just had mussels and things like that on it, and that did seem far more stable. We would see this plastic every week, go back, and it looked basically the same.
Yeah, because you have some who just once a year, and if they are there, you can actually calculate at least how long they've been there.
Yes. So definitely needed more time for this project.
Yeah, and a really nice study.
Thanks. I have one question. Well, when I was surprised that both marine estuary and freshwater biofilms in the same way, I guess, the literature on marine biofilms is very well developed. It's very long term. So how does that compare to find out what you expect, your expectations from the marine literature?
To the river. Great question. So yes, you're absolutely right. The marine literature has been a long time coming. The riverine biofilm literature, even estuarine, started maybe a few years ago. There's very limited papers out there, especially for the plastosphere, the biofilms on plastic. So honestly, for this project, I pulled a lot from the marine literature. Like the wall model is a marine epibiosis model, and maybe that's why it stood true in the marine setting and not in the riverine setting. But we were quite interested in how the hydrological conditions, like they impact the marine and the riverine the same way, and they absolutely did not. So it is kind of two different groups, and also if I could do this again, I think maybe even cutting out the estuarine, but giving the marine a little bit more of my energy and effort, and then also giving the riverine a little bit more, sample sizes and things like that to really compare the two.
So I mean, it'll be interesting to see how all develops the riverine side. And I guess the final obviously growth of these communities is going to be seasonal, especially with our rivers.
Yes.
So what do you expect to be happening?
Yes, so we knew that we wanted to sample at this very specific time. I wish we had the nutrient profiles of the rivers. In the winter, it might be a little bit more, in the summer, a little bit less. But yeah, we didn't have the nutrient profile, so we're not quite sure. But we wanted to make sure just to study.
What about just temperature?
Oh, temperature also can change, but it really depends on, again, I wish we had molecular techniques to look at the species, right? There are some species that are more tolerant to changes in temperature, just like they're more tolerant to changes in salinity. We know that some of the diatoms we were working with, just from previous work of my group, can exist all along this river, while some are actually quite specific to one spot. So the same would be probably true for temperature as they increase. In the winter, well, depends on the nutrients and depends on their tolerance, but yeah.
Are there any questions from the audience? Would you pass the bill?
Test a really dynamic, stable environment.
Yes, but I guess the question is like, where would you find this really stable environment? Of course, there could be more kind of laboratory studies done, but again, that's not as interesting as to the people that are trying to put out these active litter collectors. They would like to see it in the environment that they want to put the litter collectors in. So yes, of course, it would be great, but I think time and efficiency, we're just going to use the environment we were given.
There are lots of congratulations coming through online. No questions from the online chat. Thank you. And,
Thanks.
Thank you very much, Marie, for the assistance. So my name is Eva. This is the last but not least session this day, this room. First, let's introduce the jury. They are Victoria Carpenter and Andres Arias from University of Massachusetts. We will allow for it to second by Janelle Levy, Kathleen Nicholson, and Natalie Islam. As you know, 15 minutes for the presentation and then for the discussion with the jury. Also, if you have time, also for additional question, audience, and if you have a question, chat if there is time to make it. Yeah, okay? So, when there are only 5 minutes left, I will say like this, okay? So, 5 for you. So, just when you are near the end of the presentation, I raise the hand, and when you are like 1 minute left, I will start. Okay? This way we can have time. So, I call the first presenter, Janelle Levy.
I hope you don't mind, my notes are on my phone, so I'm not just okay, so the name of my presentation is From Beliefs to Management, Exploring the Link Between Fishers Perceptions and Coastal Resource Management. And we do have a quick introduction, an agenda. We're going to go through introduction, methodology, results and discussion, conclusions, acknowledgements, and then I do have a reference slide if anyone is curious. So, a little bit of background. Coral reefs are these really extraordinary underwater structures. They provide a lot of resources for the planet and for us as humans, but as hopefully everybody in the room knows and appreciates, they are in pretty rapid decline. So consider for a moment that some 2 million people in Latin America and the Caribbean alone are reliant on the protein that is provided by coral reefs in the region. And this is 2 million people whose lives are very, very intertwined with the coral reefs, right? And this is what's known as a socio-ecological system. So for better or for worse, the humans, these 2 million people, are a really huge part of this ecosystem. And that is really important because current research tends to neglect these 2 million people, so a really massive part of the ecosystem. When we do look at a reason why maybe the coral reef is in decline, we may be looking at things like salinity or pollution, but what we're not really looking to is the humans, namely the fishers and the people who are really interacting with it on a day-to-day basis. So the organization that I have been working with for my thesis is called the Coral Reef Alliance, and they are addressing this through perception research. So this is really asking the question, how do fishers feel about the reef? And in turn, how does that make them treat the reef? And in turn, how does that affect the actual health of the coral reef? And I was really lucky because I was able to work with 2 different data sets that allowed me to compare fishers' perceptions to the actual health of the reef. And I did this by asking the following 2 questions. So 1, do fishers' perceptions in different Honduran communities relate to reef fishery health at nearby reefs? And then 2, can demographic and perception data be used to inform reef fisheries management decisions? And so how did we do this? We were looking at a 250-kilometer stretch along the northern coast of Honduras, and we were looking at these 5 different communities. This area is characterized by sandy beaches, rivers, bays, coastal lagoons. And one very important thing is that reefs are very abundant here. So everywhere that you see one of these little red dots is actually an underwater survey site of a reef. There are a lot more, but these are the ones that we included in our study. And finally, the coastal communities here are really reliant on fishing for a number of reasons, for the actual protein that they receive, because they're, as their income, as part of their heritage, so very reliant on fishing in the region. And then, as I mentioned before, we have two data sets. So the first data set was provided by the Healthy Reefs Initiative, or HRI, and that's the fish biomass data set that you see over there. And this was done by underwater visual surveys, so they went down with their scuba gear and looked for fish and documented the biomass. And this is actually really cool because we ended up with two variables from this data set. So the total average biomass, which as it turns out is a really great proxy for health of the coral reef. We also have commercially significant average biomass, which also is pretty fantastic. So this is just the fish that are actively being fished by fishers, only those species. And this is actually a pretty good proxy for reef fishery health. So we have those two. And then the next thing is the fisher surveys. And this was done by my host organization, the Coral Reef Alliance that I mentioned. And the first thing that they did was they called people and they went down to the docks and they asked people questions about demographics. So how old are you? How many people in your household? Things like that. The other thing that they asked about were perceptions. And they gave people a 5-point Likert scale with a series of statements and asked them to rate how much they agreed or disagreed with each statement. And we did divide it into 3 categories. I'm going to come back to that in a moment, so don't worry about that. We're going to return. And then the very last thing that we did was we grouped all of them. So as you can see, we kind of just went visually and looked at these and said, okay, here's the community. Where are the closest reefs? And then we grouped them together. And then once we had that first map, we took it to some local stakeholders and we asked, is this where people fish? Do we have this right? And we got confirmation that yes, this looks correct. This is where people are most likely fishing. These are the communities, and this is where they're fishing. And so this is the map that we ended up with. And before I move along, I wanted to return to what the actual survey looks like. So over here, we have our 3 categories: emotional, critical assessment, and observational. These are all the different statements. And as you can see, they have a 5-point Likert scale where they can go all the way from the very sad face of disagreeing all the way to the very happy face of strongly agreeing. And the way that we categorize these, for example, we have one of these statements is, I feel very emotionally connected to being a fisher. So something like that would go in the emotional category. Critical assessment, a little bit harder to define, but what this is really looking at is how do people feel about how the reefs in the area are treated? So how are the social systems, how are the governance systems doing? How are they treating the reef? This is sometimes called social capital, you might see in the literature. And these are going to be statements like the different operators, such as NGOs and tour operators, get along in this area. So kind of just questions or statements about how are the resources here being treated. Observational, this is the final category, and these are going to be the very, very tangible things. So for example, every time I go fishing, I make a profit. It's a very, very tangible statement, and so anything that was super tangible like that, we just grouped into observational. All right, so our analysis. We started by grouping everything with QGIS, as I mentioned. All analyses were carried out in R. We did an exploratory analysis of both of the data sets, and we started with the descriptive statistics, and then we moved into just checking for whether or not there were differences between groups. We wanted to make sure that there were differences between the variables before we moved forward. We also looked at the internal consistency of the data using a Chromebox alpha test. And we ended up with 2 predetermined thresholds. So the first one was the kind of classic threshold, alpha of 0.05. But because this is really just exploratory, we also wanted to include a 0.09 alpha because we didn't want to go ahead and exclude results right away that might be significant later. And then particularly with the perception data, we grouped these by the 3 categories that we mentioned. So all of the tests were grouped by critical assessment, emotional, and observational. We also changed the responses to just 1, 2, 3, 4, and 5 so that we could work with the data continuously. And then the last thing that we did was compare them using a simple linear regression. And so we just compared all of the different combinations using the simple linear regression. And our first key finding from the exploratory analysis was that the level of education is really significantly associated with being a fisher. And what we found here was an inverse relationship. So the more educated people are in a community, the less likely they are to be fishers, the less fishers there are in that community. And you can see this all together, and you can also see it just in the individual communities, with the exception of Trujillo Santa Fe, but altogether this is quite a big trend. And this is actually a really great piece of information when we were looking at making management decisions, which is the end goal. We'll come back to that, by the way. And then the next thing that we're looking at is perceptions. So these are our three categories. We have critical assessment, emotional, and observational on this axis, and then we have the ratings on the y-axis over here. And I wanted to put the p-values up here because I wanted to show that these are very tiny p-values. And what that means is that we did a good job of dividing these up into very distinct categories, right? And when we really take a moment to look at this, what we're seeing is that in the emotional category, people feel a very strong feeling towards this. So the average is way up at the top, right? So people are telling us that fishing is very, very ingrained in who they are as people in every community. This trend is across every community. Critical assessment, so if you remember, that's kind of social capital and how people feel about how the reef is treated. People seem to feel moderately satisfied about how the reef is treated. And then the lowest is going to be observational. So these are, questions like how I make a profit every time I go fishing, and we're seeing that people are not particularly happy with that. All right, so what does this look like when we compare them? So the first thing that we compared was the total average, and if you remember, we use this as a proxy for overall reef health. And then we have the Likert scale ratings on the x-axis and the biomass on the y-axis. And then they're divided up. Each one of these lines is one of the categories. So we have critical assessment, emotional, and observational. And, excuse me, our million-dollar question is, which one of these lines shows significance? And for this one, the answer is, we saw what we were calling marginal significance in the observational line. And what does that actually mean in practical terms? Well, it means that there is some evidence to suggest that fishers are seeing declines in the reef. Oh, go ahead. Oh, 5 minutes. Sorry. So that, in my opinion, is not a very interesting result. So I'm going to go ahead and move on. The next one that we have is commercially significant biomass. So only the fish that are being fished. And as we mentioned, this was a proxy for reef fishery health. And where did we find significance here? This is much more interesting. So we found that the line for emotional is, the p-value is 0.051, which is frustratingly close to being significant, but thank you, science, for landing on marginal significance. And then critical assessment, this is significant. So we have found significance. And I wanted to take a minute and just talk about what that actually means. So with the critical assessment, we know that the happier fishers are with the social and governance systems, the more fish there will be. And we don't know if that's because if they're happier, there are more fish, or if there are more fish because they're happier. But what we do know is that when we're making management decisions, the critical assessment or the social capital is going to be a really good first place to start looking because we know that that has the most effect. And similarly with the emotional category, we don't really know which way this goes, but we do know that when we're trying to appeal to fishers, we should appeal to the emotional side if we, want to have a good starting point. All right. And I wanted to circle back to my original questions just to kind of talk about, did we answer these questions? And the first one is, do fishers' perceptions in different Honduran communities relate to the reef fishery health at nearby sites? I think this is, yes, absolutely. We found significance here. But what we did see is that not all perceptions are created equally, right? So some of these categories are going to be of more use to us than others. And then the second thing that we saw, the second question is, can demographic and perception data be used to inform fisheries management decisions? And personally, I also think, yeah, absolutely. Just from the short analysis, we were seeing a set of communities where people have fishing really deeply ingrained in their identity. And this has a really big implication because one of the major management techniques there right now is to try and pull people out of their career and put them into a new career. And why would that be successful when we have, people who are so connected to fishing emotionally? So maybe a better option based on this data is to connect them to a career that is somewhat similar to fishing, such as what Katie's going to be talking about in a minute, or maybe focus on educational opportunities because we did see as well that education has a lot to do with people, the amount of people who are fishers. So we definitely see significance here. And then just to jump into conclusions really quickly, perception research can be really useful, right? So in my opinion, this gave us some really great insight and direction that can actually be used down in Honduras for making decisions about the fisheries there, right? I feel that these results really show the importance of using the human dimension and looking at the human dimension when we are trying to make these decisions. And if we're not looking at that information, then we are literally missing out on the opinions and the thoughts of 2 million people, which again, not a trivial amount of people, who are contributing to the ecosystem and who are a part of that socio-ecological system. And the final thing I wanted to do really quickly is give some acknowledgements to the Coral Reef Alliance, who was kind enough to give me data, also the Healthy Reefs Initiative, who gave me a fantastic data set as well, Antonella Rivera for her supervision, and very lastly, my fiance, who flew all the way here from Texas to watch me present. And here are my references if anyone is curious. And thank you everyone for listening, and I can take questions. Thank you. Yeah.
I just wanted to say also, like, I'm from University of Washington, but I have a question. First of all, thank you for the presentation. I really find your topic very interesting. It's also a little bit of a slap in the face for me because in the sense that when we are studying marine ecology, as me, I'm very language research and practical ecology. That's why I do it. But it's obviously that we have to take into account when we think about fisheries is bad, and very, very quick to say stop. There's people behind it that depend on that for a living, and it's even worse is the case in South America. So it's really nice the way that you put it, and the intro was really, really interesting. I wanted to just tell you that you can skip a little, you can get a little bit of time. You don't have to talk about your agenda. You know how it's going to be. So that you can just leave it, and next time prepare this lecture.
There's so much information.
That gives you a little bit of extra seconds to read it. Because you did a really good presentation. Obviously, your English is great. I think it's your first language. Yeah. And then I was going to tell you just to be a little bit more mindful of your audience sometimes, because obviously your pronunciation is great, you go fast. So when you know that your audience is not, mostly are not from an English country, but it was fine. I get it. A little bit sit there. Then I think you presented a lot of stuff. And I had a lot of things to discuss, so we had to prioritize, but then I got curious about a few things. I want to know if you're involved in the making of the surveys, the survey, if you were the person that wrote questions, or
Oh, no, so I wasn't involved in that, but I did do, I was the person who was like dividing up the categories, so I kind of like divided up and figured out the analysis, but I wasn't involved in the actual making of it. I was so lucky that I just got to work with like the 2 data sets, and they have these like, oh my god, they were so great, like just really 2 beautiful data sets that I got to work with.
And then about the data set where you had the biomass, I wanted to know any info as biomass species identity. Fish, or it's just total fish biomass and that's it.
Yeah, so the way that the data set is organized is there are different columns for every species. And I believe that it was fishbase.org where you can go and just see which ones are the commercially significant ones for that area. So we just went and found the commercially significant ones and then added all the columns of those species.
And then another thing was the educational level. I actually quite grasped how we got to that.
Yeah, totally. Oh, I went too far by a lot. Yeah, so we actually had, I had to jump through this really, really quickly, but one of the questions in the Fisher surveys were demographic questions. And one of the questions was, what is your education level? And so we had all kinds of different answers, but we managed to put them into 4 different boxes. So there's no education, primary education, secondary, and higher. And so this is what you're seeing. There's none, primary, secondary, and higher. And then in every one, you're looking at the reddish is if they're a fisher or not, and then the blue is if they're not a fisher, right? So in every one, you're just seeing a downward trend, like the higher the education, the less fishers.
Okay. No, thank you. I was just a bit confused. I thought you were only serving fisher.
Yeah. Yeah.
So you weren't. It was the opening.
Yeah. So the way that they did it was the survey asked a lot of demographic questions, and then at a certain point, if someone answers that they are a fisher by trade or they do fishing as some kind of occupation, then they continue on with asking about how do you feel about the fishing industry. So like the entire data set has a ton of data about just like they wanted it to be really encompassing of the community, but then they only wanted for the not the demographics, the perceptions, they only wanted the perceptions of the people who are actually really like involved in the fishing.
Thank you.
Thank you, and thank you for feedback. Hello.
Thank you. Nice presentation, but we have to comment. I would like to see in the presentation also the conclusions that were not mentioned. So, take your opinion somehow. But I think that also is important for the audience. But this is an academic work, so we should have the choose even with numbers.
Okay.
So we do have 2 questions that would be true objective. I don't know exactly. Do you aware of that? I mean, you see the
Yeah. On the conclusion slide, you're looking for just something like bullet points. Want to be able to see like the yeah.
Suppose that this should be in concordance with the objective work as well. I mean, this is structure and I miss this structure. About that, here, and it's difficult to make questions. So, it's right. It's nice, it's difficult to evaluate something and that they know exactly how much for things that work is done.
Yeah, so it's a lot of information. So I apologize. I know I talked really fast. So
yeah. And I have a quite complicated question. We were in the was quite common the question about the committees of ethics and this kind of certificates and that we you know if it was requirement or something to feel for the institution because I'm telling you that because here if you want to make this you have to pass the M10 in your case that is sometimes difficult.
Yeah. So I did not have any involvement with the ethical parts of it because this was just I had the chance to use the 2 data sets. What I do know is that particularly the Coral Reef Alliance, who I was working with, their surveys were voluntary, so I'm not quite sure where like an ethical consideration might come in just for because people have the option of declining, which could put in a sampler bias, but they did also have the option of declining. So I'm not sure exactly where the ethical considerations would come in for that. However, I would be curious to know, and I don't know this off the top of my head, but the other ones that did the underwater visual surveys, that was actually a third party, but they have a really comprehensive paper with all of their methods, so that's something that I could potentially look up and figure out.
I mean, because that's quite the student have to feel something.
Interesting. Okay.
If not for them,
Yeah.
it's just speaking tradition. Also for patients.
Okay. So that's good feedback. I can't say I really ran into anything while I was doing this, but that's really great feedback. Yeah.
It's understandable. But yes.
Yeah. Especially because they're so different across all different countries, and we are like an aggregation of so many countries. So, yeah, that's great feedback. Thank you.
Which was your objective? Clearly, one sentence. What is the objective of it?
To understand whether or not looking at someone's perceptions does really have tangible effects on the coral reef, if I had to put it into one single sentence.
Okay, and knowing that, the title we have is appropriate or not? Okay, we have another one.
Sorry?
We have another title.
Oh, yeah, so these are preliminary results. I haven't turned in the actual thesis yet. I'm sorry if I forgot to mention that at the beginning, but yeah, I have changed the title since, and when the final manuscript gets turned in, this will be the
title. Okay, so I think it's
perfect. Thank you. I appreciate it.
Okay, thank you for your presentation of aims and also the scope of your work is very useful. I also concur with the comment please just speak slowly because sometimes you have a lot of ideas on the same slide. Was very tricky sometimes to follow. Well, I have a question. An example of how this type of studies can help to write laws or write laws for management for fisheries.
I actually did not come across any studies that were used for that. One thing that I really did find is that the literature as far as perception research is, it really seems like it's in its, I had a lot of trouble finding things that were directly relevant to, there is kind of a growing body of literature on perception research, but very little when it comes to like ecology or coral reefs, so maybe it's out there if it wasn't.
So it has a great potential for helping the government.
I think so. Yeah, in my opinion, some of the things that we cross are yeah, absolutely. Which I thought was really exciting. When I came into it, I had no idea where to find. Like, the more that I was looking through it, and the more I was like, you know, when you're looking at the charts, there is really just like a linear relationship, and I was like pretty shocked, and so I was kind of excited to see that there are actually conclusions to that. And yeah, very exciting.
And also, when biomass, you mean it has a commercial value?
Yeah, so we have two different. So these were the two different variables, and the first variable was just total average biomass, so that's everything all together. But then we also had just commercially significant biomass, so that was only, according to fishbase.com, the species that are considered commercially significant. And so this is just those species. So if you look at the two graphs, one has like a very high number, and that's the total biomass, and the other one is like relatively low, and that's just the commercial.
Okay, thank you. I'm congratulations.
Thank you. Thank you so much. I appreciate all the feedback.
So just a quick question because I want to give time about this part of biomass and all that. Because I was wondering if you have like this preliminary results kind of already. I was wondering what are you planning to do? So for your handling and taking more analysis on this part.
I think that these are the main results. I don't think that I was planning on going too much further into I'm not really sure if I understand your question. I'm planning on looking further into.
It was a big thing because I was trying to think about like the pros and cons of using just biomass as a system health. Because like if you tell that to most ecologists, they will
Okay.
It's not the only thing, right?
Yeah.
Biomass tells a little bit of the story. So it says like a system health. It's not see if you had that idea, if you have any other ideas of what you could use with that data to get maybe other businesses or something like that.
Yeah. So these ones were two just particularly convenient things that are just like here and like in perfectly exactly the format that we did. There were a couple other things that I ran into in the literature. So for example, parrotfish, there's like a direct relationship between how many parrotfish there are, and if you take a certain amount of parrotfish out of the ocean, then suddenly there's like this rapid decline. So, and I think we do have like the column just for parrotfish, so that is something that I can take a look at. So yeah, there are a couple things that I ran into in the literature, but I think this is going to be the bulk of it. Yeah. Yeah, good question.
Thank you. We have only there's time for one.
What's up? No, I did not, and that's something that I wanted to be able to look into just a little bit more, but it would require kind of a lot more analysis. So that might, like, depending on kind of how things shake out in the further analysis, that may or may not be something that I include. Yeah. Cool.
Okay.
All right. Thank you, everybody. Yep. Thank you.
All right, good afternoon, everybody. Thank you so much for being here. My name is Catherine Nicholson, and today I'll be presenting to you about small-scale aquaponics as a livelihood diversification strategy in Honduras. And as you may note, this is a different title from the one that you may see on the program, and that is because, again, this is also a work in progress, and this will be a provisional result today. So a brief overview, which I'm not going to go into because it's pretty standard, but that's that. So a bit of background, again, this is my title, and it kind of describes the what, which is livelihood diversification strategies. How that's going to be implemented is with small-scale aquaponics, and then the study site describes the where, the who, and the why. So going into livelihood diversification strategies, which I will refer to as LDSs from now on, just for convenience and time, are referred to attempts by individuals and households to find new ways to raise incomes and reduce environmental risk, according to Hussein and Nelson. And as Janelle stated earlier, it's difficult to pull fishers away from their livelihood, but this is more focused on diversification rather than an alternative. This would be supplemental and additional. LDSs also contribute to community empowerment and resiliency by adding additional sources of income that can be stabilizing during volatile events. And they are also considered a necessary component of improving reef system health by lessening pressure on fisheries via reduced fishing effort. Finally, tailored project design and careful beneficiary selection are crucial to LDS success. Okay, a brief overview of small-scale aquaponics, which is the LDS that is being proposed. They basically, it's a combination of aquaculture and hydroponics, which is growth of plants in a soilless culture. It combines fish, plants, and nitrifying bacteria in a mutually beneficial cycle and ends up with products of plants and fish for consumption or income provision. Some additional benefits are its low water use, minimal inputs, expandable nature, and that materials for it can be found relatively cheaply in most areas, making it suitable for creation in resource-poor areas. Where is this all happening? It's happening in Central America on the north coast of Honduras, in the Tela Bay, which was one of the areas that Janelle mentioned earlier. A little bit more focus on who's living in this area. We focus on two communities. One of them is called Tornave and the other is Triunfo, and these are Garifuna communities which follow traditional livelihoods based on their mixed African and indigenous heritage and have a high reliance on fishing activities for their livelihoods, as well as tourism. Tela City is also of note because this is the commercial center of the area, as well as the site where the prototype aquaponics system is currently. And so why are we focused on this area? As Janelle said, it is the site of the Mesoamerican reef, which is the world's second longest reef, obviously with very high biodiversity and providing many ecosystem services, and those 2 million people relying on fish in this area, or for protein in this area, and there is a decreasing fish biomass as well as decreasing coral coverage. Focusing in on the Tela Bay specifically, all of these boxes are protected areas, as many of the reefs in this area are not yet found to be impacted by coral tissue loss disease, making them pretty special, and maybe that will change in the coming years, but at the moment, the coral coverage is relatively high compared to the rest of the reef, upwards of 60%, while the fish biomass is relatively low, which is interesting. There's a high reliance on these depleted reef fisheries, though, from the several fishing communities, which are little circles, including and finally, because there are all these communities, there's a high potential for increasing community empowerment. So some objectives of this project were to address my question, which was, what is the potential of small-scale aquaponics utilizing native fish as an LDS with reference to specific situations? Some overarching goals, in addition to increasing community resilience, could be informing stakeholders and LDS implementers in the entire Mesoamerican reef region, leading to materials that can support LDS implementation success, as well as supporting fish population recovery. And some direct objectives, which I'll focus on throughout the rest of the presentation, are describing previous LDS successes and failures and lessons learned in the area, describing the designs and components of the design of the LDS system, and describing potential beneficiary communities and analyzing the optimal fit of the LDS. So briefly into the methods, I did a literature review, very standard, so I'm not going to get into it, but it informed the first two objectives of the study. I performed bilateral interviews with the nonprofit local coordinator there in Tela, with the PI of the nonprofit and other stakeholders. These were done virtually and in person. Some of them were done on site with the aquaponic pilot system there in Tela, and more than 20 interviews were done, some short 10-minute Q&As, while some were like longer, hours-long sessions. And these informed all three of my objectives. Finally, the third method that I'm going to focus most on, because it has the most quantitative results, are the community focus groups. These informed the second two objectives, and they started with creating questions and themes. Those were the 6 themes that I'll focus on throughout the rest of the time as well, so I don't need to get into at this moment, but these were prepared in order to address the objectives and the questions. Next, the communities were identified. This was based on whether or not the communities had running water and electricity, which were necessary for an aquaponic system. And this ended up being Tornave and Triunfo, the 2 communities that I mentioned earlier on that map slide. And then the focus groups were held. These were done on site at the communities during already occurring meetings. And after having the consent of the community members, they lasted around 90 minutes and 120 minutes for Triunfo and Tornave, respectively. And finally, the transcripts were created, and then thematic analysis was applied, which I will talk to in this next slide. So, applying thematic analysis to community focus groups includes several steps, the first of which I performed was deductive coding, where I create initial codes that fit into those original 6 themes, and then manually code the transcripts through a process where there's a quotation such as one about previous tilapia aquaculture community project challenges, and then I applied positive and positive themed codes for previous fish experience or community projects, and then negative ones such as previous project challenges. These are just examples of the many codes that I created. The next step in the process was an inductive coding step where I utilized the AI software that is included in IMBRSea, which is another software program. The software created its own inductive codes, similar to those ones, but they were extracted from the transcripts themselves and not associated with any previously existing themes. They had to be verified and edited to make sure they made sense within the context. Next, I combined these inductive and deductive coding processes by sorting the newly created inductive codes into the pre-existing codes. Either if they matched alongside a code that was already existing, they were sorted under that code, or within the themes that were already existing. However, there were some uncategorizable codes that I'll talk about more in a bit with themes. And finally, I analyzed the code frequencies by comparing the raw frequencies, standardizing the data, and then comparing the communities within that, which I'll get into in the results. But first result I'll focus on is back to that first objective of describing the previous LDS successes, failures, and lessons learned. So what we learned were there were 2 previous failures and 1 successful LDS still running in the Fela area. In the failed projects, community members lacked experience and interest in the areas of those projects, which were microfinance and shopkeeping, so not related to fishing at all, and they were also tried to be implemented in fishing villages. This pointed to the second point, which is the importance of LDS design tailored to the community and incorporating areas of existing expertise and interests, which in this case would involve revolving around fish. And finally, long-term expertise support and community empowerment with a variety of tools were shown to be really important for LDS success in the area. Next, really briefly, I'm going to touch on a couple aspects of the design, such as the fish species that we've been focusing on, which are the Vieja caulicada, which is the black belt cichlid, which is this one here. This one is kind of younger one, and then the IMBRSea ophthalmus, which is the Mayan cichlid, both of which were deemed to be desirable for consumption, especially by the Garifuna communities. They were very excited about the black belt cichlid. This one, they're both compatible with an aquaponic system. They can grow and thrive within the system. They can be part of a sustainable system in that they can actually reproduce within the system, and you can keep the fish cycle going for a long time within that. And finally, they are native species to the area, so there's no problem if there's any escape. Next, some plant species that we use in the system were habanero peppers, tomatoes, and cucumbers. These were deemed desirable by the community members. They're compatible with the system. They're relatively resistant to pests, and there's even the potential of creating added value products such as salsas with some of these, which could add additional income. Oops. Okay, into results and discussion. This is a graph of results and discussion of the potential beneficiary description. So this is a graph of positive and negative code frequencies and total code scores for each theme for the 2 communities. Some things of note were the relatively high values here, and mixed values for the orange community, which was Tornave. They described a lot of previous projects that they did as a community and some of the challenges, successes, failures, things they learned from it. So that contributes to those relatively high scores. The really low scores for pre-existing assets and resources might seem concerning, but it was based on we asked questions about that, and they responded very quickly that what was needed was already existing. So there just weren't very many mentions of that. And finally, interest was at the highest code frequency for both communities, and that shows that this kind of project is really of high interest for these communities. This next graph is showing the standardized proportions of positive and negative score frequencies among the 6 teams, as well as a combined total for the 2 communities. And as you can see, they're very close, but the Tornave, or orange community, is slightly higher than the other, and that along with previous anecdotes and results from interviews has us determine that the Tornave community will be the optimal community to have as the first pilot community. So one more aspect is looking at the uncategorized inductive codes. These were some codes that were created by the AI that didn't fit into any of the code categories or themes. And from these, we could extract two emergent themes. The first one being commerce, which is really of note because often in environmental implementation projects, we often aren't very focused on commerce, but this is obviously of great importance to the communities. So we should focus on that going forward with the creation of a business plan. And second was the environmental awareness emergent, which is also really of note because it indicates that environmental awareness is important to the people. They're interested in conserving the environment, and they know that there are environmental issues, which is very important, especially when trying to implement these environmentally themed projects. Okay, on to main conclusions. There is a strong need for suitable LDSs to reduce fishing pressure in the Mesoamerican reef region, and small-scale aquaponics appears to be a really viable option based on the reception by the community and the match of the LDSs to the community's needs. Tornave is the more optimal community as the first model because of their previous experiences and high interest, and there are opportunities for expansion at the same time to further communities. And finally, environmental awareness is highly relevant for the communities and within the project. A couple of future directions are the creation of community empowerment tools such as workshops and manuals. A business plan should be created and adopted, and the model should be expanded. Finally, polyculture potential. There's a potential of adding further species to the aquaponic system, such as shrimps, for further income. So basically, when applied with a careful understanding of the community's needs and interests, a well-designed livelihood diversification strategy can be a part of making a difference in community empowerment and the movement toward more sustainable fisheries. Here's my references. Thank you all so much. Any questions? Thank you.
Thank you for the presentation. I think that your presentation on the whole is very interesting to work with the marine environment and everything.
Yeah, very
so it's very important that think about preserving some marine environment the marine way.
Yeah.
With all the activities. It's very, very interesting. So congratulations. I saw that the species of fish are also freshwater species.
Yeah, they're both freshwater, but they can survive quite well in like pretty saline situations. The lagoon that most of the fishing happens in this area, or that a large component of the fishing happens in this area, is connected. It's a saline, relatively saline lagoon, so they're tolerant.
And I'm also calculate the space, the physical space for the tanks you need for
Yeah, that's another part of the aquaponic system design that I haven't finished investigating yet, so I haven't presented it here. But again, that will be hopefully included in the final thesis work. As I'm also trying to include like a cost-benefit analysis type of thing, including like the electricity inputs and water inputs and things that are necessary for the final function of the system.
So the people from Tornave have the facilities and the space for
yeah. Yes.
And also the podcast that use a great literature for your study. And when you work with great literature, you have to check it carefully because it's information and check there.
Yeah, true.
So
some of
a process for
most of the gray literature that I have used was maybe from previous IMBRSea people or things that the Coral Reef Alliance, the nonprofit I'm working with, has been using, and is data that they are finding valid. So that's most of the gray literature that I'm using on, but it is a good cautionary note, of course.
Okay, thank you.
Yeah, thank you. Thank you.
Thank you. Next.
So thank you for your presentation. It's really interesting. I've never heard of this before, and for me it was like amazing, really. I think it's very, very important work, very needed. And again, as I said, the last saying we need more people working on that, and it's really like a lot of work. It seems like a lot of work, and I think sometimes I was a bit surprised that this is preliminary work, honestly, because I feel like you've already showed a lot. So you're doing really good. I think it was really, really clear because your objectives very clearly. The conclusions were really a lot more in alignment with your objectives. So very good. I thought that the thematic questions that is really interesting.
Yeah, really interesting.
For me, it's I made a parallel with what we do in product development on the method. And when you talk about functional diversity, we kind of have the same ideas there. You have the coding, you do fuzzy coding as well in terms of categories of traits, and also the whole concept that you showed really reminds me of what we have functional diversity in an ecosystem. When you have more species that have different traits, the ecosystem usually is more resilient.
Right.
And it's kind of the same thing that we have in the species.
Yeah, if you have more livelihoods, you have more.
So maybe, I think both could draw inspiration from each other. It's really, really cool the way they present it. It's very cool as well.
Thank you.
So just really big job and very interesting work.
Thank you.
Yeah. I think that the initiatives were all very well.
Thank you.
Okay. I can be the bad. Because I agree that there is a lot of work, but I don't know if it's well focused. I will just my mistake. For me, the third objective is the one that so maybe it was not that.
The first two? Yeah, I considered that in creating this, but I felt like they were important parts of the research, and they were initially the main focus of the research, and it kind of meandered and came to be focusing on the focus groups and the thematic analysis, which wasn't expected from the beginning of the project, but because where it evolved to, so I guess I was kind of reticent to get rid of the first components, but I see what you're saying and that could be it kind of was the main focus, but that could be the only focus and still, and maybe be more straightforward and clear as well. That makes sense.
Nice introduction to understand better that, and maybe should be nicer because also you could explain better.
Yeah. With the limited time, it was hard to choose which specific things to focus on.
Okay.
But that makes sense.
That was somehow frustrating for me because I like a lot of people. So
I know, it was so short.
I mean, because it's not. And I'm talking about the money, and I'm talking about the fresh water animals. What is the opinion of the people? I mean, they prefer that fish than the marine ones.
I was kind of surprised about this also because in the main city of Tela, these freshwater ones are not the most popular. There are some of the ocean ones that are much more sold at higher prices and are more valuable, but it was very pleased, and we were surprised in the focus groups with the Garifuna community that they said that their most preferred fish from the ocean or lagoon was these freshwater fishes, and they said that they were like the most culturally interesting and tasty fish for them. And I was like, that's honestly great.
It's really nice topic.
Yeah.
Well, socioeconomic and probably because in Basque and we like this food. I mean, it's not. And we have some experience in Basque country that is going to have some experience of that. Because that this wasn't that.
Yeah, that is. There's not aquaponic here right now, but there is aquaculture all along the coast, and it is tilapia, and everyone's like nobody likes the tilapia there, so they were excited about the potential of using this other species.
And what is more productive, the fish or the vegetables?
Good question. I think the fish are what is more exciting, and hopefully what will draw the fishermen to this project. But the vegetables grow faster, produce more, and usually end up being more economically productive. But I think the fish also grow relatively fast. But in general, in aquaponics, in the literature, usually the vegetables or the plants are much more lucrative.
Okay. I taste some lettuce for aquaponics.
Nice.
I have one more. If you introduce the data coming from electricity, that is
still feasible, like
at least
yeah, I'm looking forward to seeing that, but I think that it will be. It really doesn't require very much water input because it's a closed system, so it requires some if you clean and some from evaporation and evapotranspiration, but it doesn't require very much water. And in this part of Honduras, there's like a minimum level of electricity which is paid for by the government. So if you use under some certain amount of electricity, you don't have to pay, and there's very low levels of electricity use already in these communities. And with the prototype system, that one currently uses so little electricity that it doesn't have to be paid for.
You see, this is stable.
A power off time. That's true. It has been relatively stable. That hasn't presented an issue for the prototype, which has been there for the last 3 years. So far, so good.
Okay. Thank you very much.
Yeah, you're welcome. Yeah. Yeah, so I wanted to combine the deductive thing and the inductive process. This was based on some of the other people who have applied thematic analysis. Other people have combined the two processes. Some people have done just one or just the other. But ideally, if you're doing deductive coding, you can have a bunch of researchers working on it to avoid researcher bias in it, because it's just me doing it. So I wanted to try to combine another process, and then I didn't want to use just the AI coding by itself because it is a relatively novel tool when applied. As some other researchers have done, I wanted to combine the two types of processes to get a more robust result. Yeah.
Approval committee of research because it's the necessary
right.
So the country, but even for work, because it's masters and so we must follow the peer course. So it's just a general trying to or explain very well that they were informed about and especially 100% any
Right. Everything is delabeled and yeah. I should have added that, but yeah, all the right. Everything is de-identified and will continue to be. But yeah, I'll seek if there's an easy way to get post approval from a European institution. That's a good idea. Yeah. Thank you.
Thank you very much.
Thank you all.
Thanks.
Hi everyone, my name is Mazharul Islam and I'm presenting my master thesis titled Mussels as Ecosystem Engineers, Effects on Seabed Dynamics and Surrounding Ecosystems. I did my master thesis at ILVO, the supervision of Dr. Hart Franco and Alexis Miller. So we all are aware of climate change and the significant challenges it poses to the coastal communities worldwide. Engineering organism, for example, saltmarsh, coral, mangrove, oyster, and mussels can be a potential option to mitigate this effect. These organisms are considered as ecosystem engineer as their activity or activities alter the physical state and resulting creation or modification of habitats and influencing surrounding ecosystems. So in the Belgian part of the North Sea, Coastbusters project tried to implement ecosystem engineer by establishing a mussel beds by following traditional aquaculture technique. So the first finished project, Coastbusters 1, gave some insight about the mussel bed development and their impact on ecosystems, but limitations in sampling technique and lack of long-term data has hindered the establishment of concluding evidence regarding its impact. Therefore, this study with multiple sampling techniques and long-term data aim to contribute to the understanding of the role of mineral as an ecosystem engineer under different hydrodynamic conditions in the Belgian part of the North Sea. So, Belgian part of the North Sea is a shallow sea, which is characterized by subtidal sandbank and varying level of sedimentary gravity. So in our study site is located in the western side of the coastal town Dipane, where two different hydrodynamic conditions, an exposed site and a shelter site was examined. And this exposed site is situated in front of a sandbank, so very open to the hydrodynamic activity of the open sea. And shelter site is situated at the lee side of the sandbank, which is providing shelter from strong wave current. At each side, a backbone long line and dropline aquaculture setup was installed. The idea is that the mussel larvae will attach to the line and grow to a certain size and amount. When they will grow too heavy to be held, and they will fall down to the seabed and will form a mussel bed. So this area is called reef development area, and there's a reef survey area surrounding the reef development area to monitor the wider impact of installation. Finally, there's a reference area which is far away from the installation site and no impact of the development area is expected on it. So here's an overview of the sampling campaigns and installation based on before-after control impact design. So microbenthos and sediments were collected on a fixed season once per year, and video sampling and bathymetry data were collected seasonally. And all the data were collected before sampling and after sampling. So therefore, this study allows us to compare three different factors. One is factor area, which compare impact area against the control area by keeping an extra pitch level. And factor site, which compare exposed site against the shelter site. And factor campaign, which indicate the sampling times T0, T1, T2, and T3. So we set up two hypotheses for this study, and the first hypothesis is that selected study sites is supposed to differing in hydrodynamic conditions, differing in seabed morphology, sediment characteristics, and microbenthos communities. And the second hypothesis is that the presence of mussel patches or beds affect the seabed morphology, sediment characteristics, and benthic community characteristics. So within the sampling protocol, macrobenthos and sediment were collected using Van Veen graph and data were analyzed in the lab of ILVO, and seabed morphology was observed using video transits through driving per area. Additionally, a multi-beam system was used to collect bathymetry data, and this part was analyzed in the bridge. So muscle bed persistence was observed based on video snapshot data. Changes in elevation beyond 2 sampling capacity was used to calculate erosion and deposition data, and sediment characteristics were analyzed based on Malvern master sizes 2000. And changes in macrobenthic community was analyzed using community structure, community composition, and community function. Additionally, a principal component analysis was run to observe the changes in environmental variable across different sites and habitat. Finally, the impact of muscle patches, hydrodynamic conditions, and their interaction on seabed dynamics and macrobenthic communities were examined using a linear mixed model. In this model, categorical fixed effects were area and site, and categorical random effect is campaign. So here's an overview of the topics that I will be discussed in the session. Within the seabed dynamics, I'll talk about muscle bed persistence, seabed morphology, and sediment characteristics. And within the macrobenthic communities, I'll talk about community structure and community function. So the presence and formation of muscle beds, rock patches, along with other dominant habitats such as lineages and shell cover were monitored throughout the study time. So control area of the exposed site was mostly dominated by sand and shell mixed sands, whereas impact area of the exposed site had a small amount of muscle patches, specifically during the second and third year of the installation. On the other hand, sheltered area is mostly dominated by sand mixed shell and limestone patches, with higher amount of muscle patches in the sheltered impact area compared to the exposed impact area. A temporal pattern can also be explained with increasing of muscle patches from summer to winter and decreasing to a negligible amount by the spring. So in 2020, at the starting of the project, the whole seabed, both in the exposed and sheltered area, was mostly dominated by sand and shell mixed sands. And within the 2 years of the installation in 2022, a substantial portion of the seabed had been colonized by either lineage or muscle patches. So here in the figure, we can see the changes in elevation between before and after the installation, and which indicates the impact of muscle patches and hydrodynamic conditions on the seabed morphology of the area. So erosion or deposition was mostly dominated by hydrodynamic conditions, with considerably higher amount of erosion in the exposed site compared to the sheltered site. A local impact of area was observed in the impact area of the sheltered site, with a small amount of muscle patches, a small amount of sediment erosion in the impact area. However, these changes was not statistically significant to confirm the impact of muscle patches on sediment erosion and deposition mechanism. Here, green color is the sediment erosion and red is sediment erosion. So site had also impact on sediment composition, with significantly higher amount of mud and finer sand, total organic carbon in the sheltered area compared to the exposed area. However, area didn't show a lot of significant impact except for very fine sand, which was significantly higher in the impact area compared to the control area. So the newly created muscle patches or beds, hydrodynamic conditions, and the resulting changes in seabed dynamics are likely to impact macrobenthic community in the Coastbusters area. In total, 156 macrobenthics were recorded, which were mostly dominated by polychaete, oligochaete, and bivalvia. Oligochaete were mostly abundant in the sheltered site, and which was almost absent in the exposed site. An impact of area was also observed on relative abundance, with slightly higher amount of polychaete in the control and edge area compared to the edge and impact area compared to the control area. A temporal pattern can also be explained with an increasing of bivalvia over time, concurrent with the decreasing of polychaete. Differences in density, biomass, species, and diversity indexes was observed through the different sites where all the indices were higher in the sheltered site compared to the exposed site. However, there was no impact of area on community structure. Non-metric multidimensional still shows differences between exposed and sheltered site as well as between campaign. However, nothing between control is and impact area. Simple analysis indicates that sheltered site is mostly dominated by Olivicrid, Siratilida, and Lenichicontilega. On the other hand, exposed site is mostly dominated by NCC and Megalona stone. So site had also impact on functional richness, evenness, rough river, and biodiversity potential with significantly higher value in the sheltered site compared to the exposed, but nothing for functional divergence. Area also had some impact, especially for functional richness, with significantly higher value in the each area compared to the control area. So Fuji correspondence analysis based on abundance data shows there's a distinction between the sites sheltered and exposed, where exposed site were mostly characterized by species with sediment position greater than 10 cm, exoskeleton, suspension feeder, and borrower. On the other hand, sheltered site was mainly characterized by direct level development, asexual widening, and living habit crevice, hall, or understone. However, there was no differences between control age and impact. Principal component analysis shows relationship between macroventus and environmental variables, where there's a distinct differences between sheltered and exposed site, with sheltered site mostly explained by finer sediment, shell cover, lentil cover, or erosion deposition. On the other hand, exposed site is mostly explained by grain size, medium sand, and coarse sand. The presence of muscle patches, varicose sand, gravel pebble, and shell cover distinguish sheltered control area from the sheltered edge and impact area, specifically during the T3 campaign. So, seabed morphology showed a complex sediment dynamics mostly influenced by strong seasonal waves, where higher erosion was observed in the exposed area compared to the sheltered area. A temporal pattern of erosion or accretion can be observed, which is coinciding with some large strong waves. Here in this figure, dorsal frequency of waves and y-axis is showing its frequency. So a season with a high frequency of waves are likely to be a higher amount of storm event on that season. Muscle patches or beds also showed a complex spatiotemporal and seasonal pattern with completely absence of muscle beds at the beginning of the project in 2020 and 21. And disappearance of muscle patches in 2021 winter and 2022 spring. So alteration in seabed and sediment by hydrodynamic conditions mainly play the role in shaping in variation between sheltered and exposed hydrodynamic conditions based on community structure and functioning, while sheltered site exhibited more diversity, abundance, biomass, and resource use efficiency compared to the exposed site. This study indicates that muscle didn't exhibit any effects on seabed and macrobenthic community, and this lack of effect could potentially be the reason that the effects of muscles are site-specific and not always higher in the muscle bed compared to the bare sediment. And insufficient density and persistence of muscle beds or passes was observed throughout the study time. It could be a reason. And there's a substantial occurrence of Lenichicontilega, which is also an engineering species and is very known for engineering activity. So it also can support other organisms and which can potentially overshooting the actual impact of muscle patches in that area. So, in conclusion, hydrodynamic condition is the factor most influencing seabed and macrobenthic communities in the study area. And this study successfully established muscle patches or beds both in the sheltered and exposed hydrodynamic conditions and demonstrated their prolonged persistence in the absence of strong event. However, muscles had limited impact on seabed and surrounding benthic communities, and it could be the reason that there's lack of permanent muscle beds. So further research and management strategies are necessary to address the existing limitations and challenges in order to achieve a self-sustaining muscle beds capable of mitigating the impact of climate change. Thank you everyone for having me here. Thanks to my supervisor and Alexia Supervisor Alexia and my promoter Hart Van Ho, who is following online my presentation. And thanks to my Ghostbusters team for allowing me to work on this project. Thank you.
Thank you very much. First one,
I would like to start this because this is like kind of, this is my domain. I'm working on the both unique species on biodiversity. So when I saw your topic, I was very excited, and I think you probably had a lot of work done. And I think it was just a little bit tricky in some parts to follow you, and I wanted to talk about that. First of all, just talking about presentation a little bit, the pictures were very small, mainly when we were presenting your areas, so that's really hard to follow then when all your other figures are also close. Try to figure out what we're talking about, and then also when 10 facts, that's because you have so many results. That it was a bit tricky sometimes to follow. I wanted you to go back to your hypothesis, and I wanted to talk about how that was looked as well. Yeah. So the first hypothesis, you. I want to use that. What is the interest there? In that question.
First one?
And yeah, and
the projects had one goal of to observe the muscle transformation both in the sheltered and exposed hydrogen. So we try to check if there's any significant difference between the factors like sedimentation, erosion, or hydrogen conditions with the sheltered and exposed site. So
yeah, I think that you have a lot of questions that you could ask, and for me, this one's kind of a weak hypothesis because we know that. There's very different hydrodynamic conditions. See that morphology is a little bit different, it's a little bit different most of the time. You can have a few cases where it could be not only related to the hydrodynamics.
But can I add something?
Yeah.
But the thing is that, these two sites are very close to the coast. So when we talk about like exposed or sheltered site or near shore or offshore site, then we think about like very depth in offshore and low depth in near shore and very much hydrodynamic in offshore and very low hydrodynamic activity in the site. This is not the case here. It's a very close, just there's a sandbank between those sampling stations and the water depth is full in both of the sites. 5 meters. So we can clearly say that they are different. Can't say it clearly because that depth is same and the distance and both the side is very close to the nearest. It's like 2 meters and it's 5 meters. So it's not an offshore area. Our exposed site is not in the exposed. Not in the offshore area.
It's just that for me, for example, when you're trying to evaluate that's the title, right? You have the muscles.
Yeah.
And then I think that a whole part of your project was really focused on the impact of the difference in hydrodynamics.
Yeah, I know. The fact is that we didn't have any significant impact for muscles. So when I talk about something, it directly goes to the hydrodynamic condition. And because if you don't have any significant, then you can't talk about it. You can talk a lot about it.
Yeah, but my problem is not with the results. My problem really is with what the objectives really. For me, what would be really interesting would be is there a change in my response in the community when, in terms of muscle presence or not in a sheltered area and exposed to sheltered area. And I think that's what you were trying to accomplish. But the way that it's said, but I have a problem.
But it was already answered by these questions. Is there any difference of impact of muscle in sheltered and exposed? So, see my model.
Yeah, I understand that.
Yeah, if had 3 factors, 2 factors. So, it's a linear mixed model. So we have 2 factors. One is area, which is control is an empty area, and one is hydrodynamic conditions. So when we run this mixed model, that will combine both the area and site. Then you will also get the effect of muscles in sheltered and in exposed as well.
I get it. For me, you leave that. It's just the way that you present your methods that made them a little bit more clear in that sense, because then It's kind of a how do you say? We see that you thought about it, and the way that it could be presented would be like more straightforward, so it doesn't seem like an afterthought. But that's the only thing that I wanted to talk about.
But thank you for
this. You can see that you got there and you were able to speak in expression that just kind of missed the point where you could really talk about more freely about those hypotheses. And then as well in terms of semantics, it was a little bit hard to follow with the impact area as well. I got what you meant because you explained it, but it was quite fast. And then when you think about impact in terms of semantics, kind of like human impact or something like that. So it could be like muscle implemented area or stuff like that. So we wouldn't get confused because you're already talking about shelter exposed, and then you talk about impact, and it's just quite a lot of information. So try to be mindful of your audience in that sense as well. But it was fine. Like, we get it. It's just that you can make our life easier when you're presenting that we'll get to your point more easily. And then I just had a quick question to stop. I won't harass you anymore. Was that why did you use the PCA for the environmental data and IMBRSea with the formal data? What was the idea behind it? Why did you use the same strategy?
Yeah, this was done with environmental data to see the variation between sites. I mean, we know that there could be some variation between sheltered and exposed site, and also it can be between the area control is impacted. So just to confirm.
No, but my question is why the change in methods? Because you have used the PCA with the biomass, with the evidence data as well. So why do you choose to use
and IMBRSea?
Yeah.
I mean, in IMBRSea we are getting more data. From following the IMBRSea we did simple analysis to get the most contributing data that you can get from PCA.
Yeah, but then if you use the PCA, of course you don't have like the first data, but in terms of the distance of sites and all that, and the visual analysis of it, it's very interesting for community analysis. If you transform the data prior to it, I mean, I personally think it's a more informative method when you're talking about community ecology than IMBRSea. You also have that stress factor that's kind of what is it? And so just maybe if you haven't run a PCA with the community data, it could be a good thing to just check it out. Just wanted to know what was the two ideas. And yeah, anyway, it was a really interesting work anyways, and I know that the Coast Cluster project is going to be very present in the IMBRSea in the future, and you had a great contribution. Good job.
Thank you. Congratulations, and I agree with your report. And I think he's going to explore my head, which is too much. And I think that Victor was talking about that. I would like to take as well the hypothesis that I don't think that they are presenting an hypothesis. It's not an hypothesis, it's just something or suggesting that they think Victor was I would that the idea should be more focused. It's more focused. It's easy to follow. And even more, if you have clear hypothesis, you are going to perform clear hypothesis. And I think it's all that. Also, should be even better for you for explanation.
Thank you.
Not just but also just to share a lot of issues for us. Apart from that, masses. I work a lot with masses, but not stems, but as we use them for ecotoxicology. You know that the masses are disappearing some part of it. You know something about are disappearing.
Disappearing?
Yeah. You know something about the problems that there are, for instance, in Galicia, that
disease and climate change.
am looking for something related, but yeah. So all this information so far into something related to lab. How could be this information valid to try to solve the problem that we have with muscles?
Okay, I mean, we are implementing an aquaculture technique to form a muscle bed, so it can, I mean, used as a conservation purpose as well. So,
okay. And the last one, which is your contribution in all these work?
Yeah, I mean, data was not collected by me, and data analysis, I mean, lab work, I did a small portion of microbenches community analysis, and all the data analysis was done. It's a like 5 years project, so
it's not possible.
But all the analysis was done.
So I think it's clear for me. So thank you.
Thank you. Thank you for the presentation, for the work. I am a zoologist, so I would like to dive into the zoology part of your work. So I'm wondering, in Belgium, maybe after species of muscle, very close to Belgium, there is a high-risk area with high-risk species. So did you assess the identity of the species from the different shelters and
yes, when we started our project, we did types of identification, and also whenever, I mean, example benthic data, so we always get some muscle spade or juvenile muscle from this, and we identify this, and we never got anything better than those.
So what experimental studies are but sometimes the hybrids maybe stories of things, maybe, but anyways. But sometimes the difference and you find maybe not for the physical environment, but the species you have there or the process you have there. Okay. Also, I was surprised to see that before the American Raptor Plan, N68 among masses, but this Raptor Plan for soft bottoms on Zambia and for Mali. So these muscle, these Raptor Plans are humanized that are inside muscle patches or are adults?
No, it was like the bear sign.
Are in the area, not
into the muscle.
And because it was trying to be a species from the soft and also find Paleosaurs or Assyrians or Millennials, the muscles or superior on the muscle.
I mean, yeah, we found some, but it was not like any difference between like bears and muscles.
Because sometimes here the muscle muscle. Maybe the most important group are Paleosaurs, Dinosaurs, and Assyrians attached to the muscle. So, and finally, is that your study contrast another massive population are compared your results with muscle from Spain, right?
I mean, yeah, I mean, there's not a lot of work regarding the engineering with muscles, but yeah, we have found some like small work that is the like impact on ecosystems or maybe sometimes something, but not for coastal protection like ocean protection.
Thank you very much. Sorry. Well, I was wondering if I knew the 5-year project, right? If the project carried on to change, bigger issue by spreading.
I mean, it's hard to say because I mean 5 years is a long time. But I mean, we had planned. I mean, my supervisor and promoter is thinking for extending to do some social analysis. Yeah, so maybe we get something.
Thank you very much. We are going to introduce some cultural access now. Curious, and finally, I look at the world as an ecosystem. So thank you very much for the attention. Thank you also for assistance and for the great you for the thank you.